But yeah, I also like I've been watching some of his hangouts, some of G-Man's hangouts recently, and he keeps bringing me up, but he doesn't know anything about me as a person. And so it's like, I don't know what to, he's like, maybe I'll roast Scientist Sam. I don't know anything about him though.
It's kind of hard when you just like, well, he makes Kool-Aid in a lab.
Yeah, I make Kool-Aid in the lab.
That's still funny. I don't care what anybody says.
That is apparently what I do for a living. Evidently. But do you want to see why we're hosting this, or do you want to bullshit for a few more minutes until people start getting the notification? Because I just got it on my phone.
Okay, I want to wait for a little bit because...
Yeah, wait a couple minutes before we start diving into it.
Yeah. And I'm actually sending some text messages to folks too, because I think EO said he wasn't getting the notification, which is
maybe
weird.
Post it in the GDC.
Good idea. Good idea.
This actually is somewhat. I mean, we're gonna fuck around a little bit, but it's gonna be a somewhat serious hangout. Also, Christian Anarchist has apparently found the video of Neff being super drunk.
Really?
From like 6 years ago, and he's like scared. He just posted that he's kind of scared now. I was like, oh, you just never saw. I hung out when I first started having conversations with Nephilim Free. It was when he was in his drunken prime. he would just get shit canned and then go online.
And I swear he's not. Okay, he's good. I don't know. That's one of those things like Neff and alcohol. They just go together real well. Like there's no denying it. We can disagree all day long about, how much he knows, but Neff just religiously drunk. That's just a fact.
Oh, guess what papers Nestling just linked in the GDC? They're the two papers about the ribosome that we had been talking about.
Huh.
From PNAS, yeah.
Look at that. So we have access to it, but other people don't? I mean, I don't think that one's behind a paywall last time I checked.
I don't actually know because right now I'm doing a file transfer from my lab, so I'm on my university privileges right now. So I would have to do for checking that. But I mean, I don't think so. PNAS is mostly open access, or entirely.
Let me see.
I know that Nature Communications. I didn't know if it was Nature Scientific Reports or but the journal that we're reviewing is all open access. So
yeah, they
don't have an excuse for hiding behind a paywall.
Yeah, PNAS. No, yeah, that's the first link that Nestle put on there. That one's not working at all, but the second one definitely is, and it didn't prompt anything for a paywall. So it's out there. It's definitely out there. So there's more people starting to join. Is this something like movie reviews, but with journal articles? Yeah, you could say that.
To some degree. We don't usually do. We've done a couple journal articles, though. We did that one about... what was it? It was the new gene evolution.
Yeah, that was a while. Yeah, that was a while back. It was sometime last year, I think.
I don't know if it was that far back. I think it was maybe 2 months ago.
Was it? There's a lot that's happened.
It's been a lot, yeah.
Okay, so yeah, all right, more people are showing up. So let me go ahead and screen share then. That might be helpful. I'm getting my screens together. Yeah, we're going Rouge, folks. We're going Rouge. All right, this is my homework that I'm working on.
I haven't read this yet. I haven't read more than the abstract.
Oh, me neither. No, I've been like when you sent it to me, I pulled it up on my other screen and I started doing this shit. Because I was like, well, it's not due until like a week from now, but might as well, like, it's easy shit. It's only 3 variables here, so might as well. So let's close this out.
Yeah, that's the secret to college success, man, is to get ahead of the early homework. Because it's always fucking easy, and then it gives you more time once you get to the harder problems. That's a problem that my students had, is the first 2 assignments weren't very hard in the algorithms course. But once you get to the more complicated stuff, you would see that people started not finishing the problems.
Yeah, I don't see why not. Like this, especially when people are giving so many concessions right now because of the hurricane. It's the most ideal time to really get in and get a head start on doing shit. So someone has asked in the chat, can both speakers please review your backgrounds, educational backgrounds? Okay, so I am a student of biology. I currently have an associate's degree in general studies. I am in my 2nd year of my bachelor's degree in science in biology with a specialization in cellular/molecular biology. And so, Sam, feel free to let them know.
So I'm a 4th year PhD student. God, that's scary to say. At a big university in bioinformatics and systems biology, where my research focus is primarily on transcriptional regulation. That's regulation of gene expression, focusing on enhancers in the context of genetic variation and inflammatory response. I have an undergrad in bio and comp sci, master's in bio, where my research focused on developing methods for protein detection on a proteome-wide scale. I tried to like break it down into easy words, not just like my science elevator pitch. So yeah,
I'm muted.
Am I muted?
No, I was muted. No.
Oh, sorry. I was like, oh, did I just say that? It's completely muted. But yeah, I mean, we could actually, some of the stuff that I did for undergrad is pretty cool stuff in terms of being simple to understand and just a little bit of an insight. Nothing groundbreaking. Decent journals we published in. But yeah.
Cool.
I think the work I do now is much cooler, but
I don't know, man. The work with the CHO cells, I mean, how can you beat that? Chinese fucking hamsters.
I thought that was cool work, but the problem is I'm not doing that anymore. And I think scientifically the stuff that I'm doing now is more interesting. That's more interesting from like a bioengineering point of view.
Yeah, I'd probably find
I'm still doing the same technology. Yeah, I mean, the stuff I'm doing now is more scientifically interesting, I'd say. It's the same technologies, but we're doing it for a different purpose. More scientific question rather than annotating the genome. Which I think is interesting. I guess in some sense I'm annotating the genome, but not in a literal sense.
All right.
So Weird Board seems pleased with that.
Yeah, he was the one that was asking. So let's. Oh, did we put the link out there for the? Okay, no, you linked it. Okay,
I did, but I'll post it again. Yeah.
Okay, actually, I'm going to.
So this is the paper that we're actually reviewing. And full disclosure, I have not read past the abstract. I don't think you have either, right?
No, I have not. So I just. I'm actually adding this to the description. There we go. Okay, so we don't have to keep posting it. All right, so
and it's open access, which is nice. This entire journal is open access.
Yes. So there you go, folks. You can check the description or the live chat, and I put it in the description so people who are watching this after the fact, you guys can go and click on it too. So, like you said, it is open access. It's fairly fucking recent. So, people who keep bringing up these, yeah, I want to say because I wanted to make a point about that because I keep seeing people, and I say people like Neffy and some of these other guys, link stuff from like 1993 or 2000.
That that's Ronnie's thing, right? Is when he linked his thing about negligible senescence, not only was it a book, right, but it was from 1994. And we've learned a hell of a lot of the biology of aging in those years, in those 13 years. I mean, you couldn't do any of the research that I'm doing right now, just technologically. Like, you didn't have access to whole genome sequences for most organisms.
Yeah, it's like, well, if it seems like 1990s is not that long ago. Like, I remember the 1990s as a kid, but when you're talking about the technological difference and the information that we had and did not have between now and then, there's a huge gap. So stuff like that is very, very relevant. So if you're doing a paper, like if you're doing a research paper for school, yeah, something from 2003 may be relevant, but you're really pushing it here, especially if you're trying to make a case as to what's possible and what's not possible. You need the latest information because shit changes given the information that we have available to us. So
also, biology, the access to next generation sequencing technologies just changed the game, right? So a lot of research is not, it's not that it's wrong, it's just it's outdated. When you can precisely identify the genes that are responsible for things on a sequencing basis, I mean, it just changes the game for the precision with which you can understand these systems. Now, granted, I mean, it's not perfect. It's noisy data. That's why I have a job. But it's an important point that you highlight.
Yeah, because that really did bother me when people were bringing up that contention. It was just kind of like, oh, well, it's a paper. Well, yeah, you're right. It is a paper, but shit changes. Sorry. That's the name of the game. So on that note, the title of this paper, and I put a long fucking title in the actual title of the video for a reason: Emergence of Native Peptide Sequences in Prebiotic Replication Networks. So it's a mouthful, yeah, but it's very important. So if you want kind of a breakdown, if you actually look at the top of the paper, it gives you a fairly decent breakdown. Chemical origin of life. Hey, supramolecular chemistry. So this is talking about that topic that we keep hearing from people outside the field. Supposedly is impossible. Like this stuff doesn't exist, and it's yeah, this, that, and the other. Evidently it can, and according to yesterday, we have some pretty good evidence as to what happened. So I'll go ahead and just read the abstract to you. And so, folks, I do have the outside chat in my other screen. So if there is something that you kind of want to touch on, please say something. Please do, because just kind of hearing words and just repeating them is not understanding. This is not the clown car posse attempt to read a scientific paper terribly game. So it's best that you have some understanding. If you need to kind of break it down layman's, that's fine. That's why we're here. That's why we do this. And this is one of the times where people who criticize us for not having content can actually ask questions, and we can explain stuff. So I don't think that any of those folks are in the live chat are going to say anything. So it is what it is.
G-Man a text message since you have his phone number.
He has to figure out how to unblock me first from on his phone, because he got pissed off and blocked me, remember? So yeah.
Of course.
So I still have him on Skype, so I'll send him the link on Skype.
And just like tag both of them in the post, I suppose, is probably.
Oh yeah, that's a good idea. It's a good idea. Let me put this.
All right, this file transfer is going to take forever, so I'm not going to do it.
That bad?
I'll get back on campus.
Never how to fucking spell his name. It's a secret. It's like a secret code to. There we go. GT. Oh shit. Always bothers me every time I'm trying to type these guys' names in.
Yeah. I mean, how are we supposed to go Rouge if we have to spell things correctly.
You can't do it properly. All right, I've tagged both of them, so there you go. All right, so the abstract, biopolymer syntheses and syntheses, excuse me, in living cells are perfected by an elaborate error correction machinery, which is not applicable during polymerization on early Earth.
So
Wow.
I think that's just saying that a lot of the basic checkpoints in, for creating the correct proteins on an early Earth aren't present.
Yeah, and this is something that people have mentioned before. This is nothing controversial. I think even Neffy was trying to make a point like this, and it was someone else. I can't remember who.
Well,
yeah,
it's a very common. It's a very common point, right? That creationists make is like, oh, well, at the origin of life, you wouldn't have had these fully formed cells. Like, well, no shit.
Hey, where? Oh, fuck, where am I? There we go. I hate that cursor. There we go. Scientists are consequently striving to identify mechanisms by which functional polymers were selected and further amplified from complex prebiotic mixtures. Complex prebiotic mixtures. So anyone who's ever heard of the Urey-Miller experiment, they weren't trying to make life. I think I need to say that here. What they were trying to do is try to find some of the basic building blocks, some of the basic amino acids, or even just some complex molecules that would be instrumental in creating them. Hold on, wife's calling.
Yeah, just that those experiments, and it's not the be all and end all, because we've of course had subsequent experiments, but I think that's the basic sketch of what these experiments try to do. Is they're not trying to create new life, but to show that it's plausible that we could create new life. I think that's the priority, as far as I understand it. Drag, you there?
All right, there we go. Yeah, she was just calling to test out her phone. Okay. All right. So next line. Here we show the instrumental role of non-enzymatic replication, I'm stressing that for a reason, in the enrichment of certain products. So if anyone's ever taken just a intro bio class, you'll learn that enzymes will help facilitate either the making or breaking of certain molecules. It's nothing controversial, but there's energy that is released and energy that's required in the making and breaking, and you can look up catabolism and anabolism and kind of get a better idea of what I'm talking about here. But it is important to know that when we're talking about those early Earth, and with the previous statement about the where is it? When we were talking about the elaborate error correction machinery, that's another type of protein. Just proteins do shit. We don't have those. We don't have proteins that are enzymatic that can help facilitate this shit. So again, this is some of the stuff that people have a problem trying to suss out. And so if there's a non-enzymatic way of these molecules becoming more complex or breaking down or whatever, then that is the solution that is going to be most conducive for this scenario. And forgive me from trying to do things and read this at the same time, but it seems as though everyone is sending me messages now. Okay, fuck me. All right. To this end, we analyzed the complex web of reactions and beta sheet peptide networks, focusing on the formation of specific intermediate compounds and template-assisted replication. That's interesting. So I'm not too familiar with beta sheet peptide networks.
Huh?
I said I'm not too familiar with what is it, beta sheet peptide networks?
Well, let's pronounce it properly. Beta sheets.
Beta.
I learned my pronunciation from Dr. True Empiricism. But there are two main like primary structures for proteins, and that's beta sheets and alpha helices. So let me see if I can find, Wikipedia actually has a pretty good explanation and a pretty good visual. So here, I'll link it in the live chat. You're looking at the live chat, right?
Yep.
Okay, just added it.
There is. Whoop.
And you see that you get this pattern where the intramolecular bonds are coordinated between the amino acid residues so that it forms this sheet-like structure. Not too hard to understand.
Yeah, I'm trying to click on it to make it bigger. Okay, well here's one. Here we go.
That's a good one.
There we go. So now that you folks can see that, this is what happens when you have molecules. You have long chains of molecules. You'll have secondary, tertiary, quaternary structures, and then once the partial charges of those molecules start getting closer to one another, they'll start interacting, and so you'll have some of these weak bonds that'll form, and they'll form in sheets, and so that will give it a particular, how can I say this best? The conformation of these sheets will help it do its job. And so when some of these sheets aren't folded properly and they go awry, there's certain things that can happen. And people have heard of some of these cases. But anyway.
Yeah, they're key structural domains.
Yeah.
And so bad, sort of some stuff that can go wrong is in a lot of amyloid diseases. This is where you get a lot of protein aggregates. A lot of times you have errors with beta sheet production. So something like Alzheimer's disease, where you have a lot of, I believe it's beta tau protein. There's something that goes wrong with the supramolecular assembly. But yeah, it's a structural domain because these things don't just function as a line, they loop back on each other. And this is sort of the, like you said, it's a primary structure. So it's the first time that things are looping back and forming structural domains.
I'll let Netslick in here for a little bit since he has limited time. Where? I don't know if you see Tyler Durden in the chat.
Synthase. I think I could, like I said, I had a dentist appointment yesterday where I got the left side of my mouth cleaned. So yesterday I could have spoken like that with that because my tongue was numb.
I'm just trying to read it and not laugh at the same time.
All right,
so like structure
that we're doing. What's it? I'll read it. Remarkably, we find that the formation of several products in a mixture that is not critically harmful, since efficient and selective template-assisted reactions serve as a backbone error correction mechanism, namely for keeping the concentration of the peptide containing the native backbone equal to or even higher than the concentrations of the other products. That is a horribly written sentence.
Jeez.
That was really awfully written. But I think what they're saying is that when, if you use the protein as a template, right, then you can copy based on that template, right? This is one mechanism of copying things. That they can do these types of template-assisted reactions without it being too critical in order to form these initial proteins.
Wait, is it saying that the protein can replicate itself like DNA does?
I think to some degree, yeah. Which makes sense, right? Think about prions.
Oh, yeah. Yes.
Right? Prions are technically a template. I think in some ways are template-assisted reactions, no?
I'm not very familiar. They are protein-based and they make other proteins to be like themselves, and they replicate like in that fashion.
I guess it's somewhat analogous, right?
Yeah. Yeah, as far as the mechanism, well, how it propagates, yeah, that makes sense.
And I mean, they just ended. We suggest these findings may shed light on molecular evolution processes that led to current biology. So this is, and we talked about this off air, where when I had that hangout with Fuzz, when I was in that hangout with Fuzz, I kind of accused Fuzz, I think fairly, that there's some level of dishonesty going on there, or he went to a shitty conference when he said that he went to a conference and there's been no development on abiogenesis since 2002. I just don't.
Wow.
I don't find that to be an honest statement.
That's a demonstrably false statement.
And again, it's not. This isn't my field, right? But even then, just this just came up in Nature Communications, which is a fairly, I'm Googling their impact factor. I mean, that's an impact factor of 12. That's a big journal.
Yeah, I'm just curious how, like I said, I was saying off air, I'm curious how he missed that, but you know, it is what it is. So, intro. Cells use surprisingly small number of molecules, primarily nucleic acids, proteins, fatty acids, and sugars, to perform an immense repertoire of functions. Well, that's an unremarkable statement. Our bodies have developed over, hell, millions of years to be more efficient rather than less efficient. Having the less efficient one selected out kind of helps with that endeavor. So this is a fairly unremarkable statement to make, but it makes for a good introduction. Even more remarkable is the phenomenon that only small subsets of these molecular families containing distinctive backbone connectivity and chiral arrangements have evolved to be operative. Again, I think it more aligns with the first statement that yes, we have evolved, and many other organisms have evolved so that, my dear son, have evolved so that we do work in a fairly efficient way where possible. I'm sure there's all types of examples of, where things could be more efficient, but, for what we have today, you can surmise that yes, that natural selection has been beneficial in helping us to be what we are.
What sentence did you get to?
Right here, for example.
For example, today's DNA and RNA molecules contain the D form of deoxyribose attached solely at the 3 to 5 prime positions of successive backbone units, and proteins are made up almost entirely of L-type alpha amino acids connected through their alpha carboxy and alpha amino termini.
And L-type means left-handed, chirality.
Yeah, it means that it rotates when you would shine light through a purified crystal, it'll rotate the light leftwards. Oh. And so what L stands for is levorotary. Rotary. I believe that's how it's spelled. And then if it rotates light rightward, in that configuration, it's called or dextrorotary, because dexter means right in Latin, the root at least. But basically what this is saying is just, how that there's a specific biological configuration for these chemicals that isn't, a priori necessary. And so they continue by saying the evolution of these unique polymer sequences in preference to others from a huge prebiotic configurational space remains a mystery, since the absence of highly evolved genetic machinery or enzyme catalysis, most polymer condensation reactions result in mixtures containing a large number of products, typically isomers of the desired molecule, each produced in low yield. And so basically what this is saying is that there's a ton of possible molecules, but nonetheless, we use the specific ones, and it's unclear how we go towards a specific set of molecules for biology. Is that the impression you got, Nestle?
Yeah. So we need a mechanism for how we get the chemicals we need, and less of the ones we don't need.
Exactly. How did we get it so that, how did we move from the from just being able to create a ton of molecules to getting the exact biologically active ones? Bragg, are you there?
Yep, I'm here.
So we asked the question how we get the first metabolic reactions to synthesize the chemicals we need for life.
Yeah, I think so.
No, I was going to say, yeah, no, it's that is an ongoing question, because I think there's what is it? Life seems to be geared toward deconfigurations or something to that effect. Because I remember someone saying something like that, but I'm not at Org Chem, so I can't speak too charismatically about that. I'm sorry, I just want to say that.
L configurations.
L configurations. I don't want to speak too charismatically about that. So, yeah, I remember hearing something about that, and that how it seems as though, things tend toward that. And so why is it, why is that the case? And so I think that's what the whole chirality thing is about. And I think there some type of some young earth creationist site that lists a chirality problem for evolution. I remember seeing something like that ages ago.
That someone does.
Yeah.
I think Voswana also puts that on his website, the chirality problem.
Oh, that may have been where I got it from. Which
I've seen papers that address that problem pretty effectively.
All right, so let me this next part, man. Many experiments directed at spontaneous polymerization of biomolecules have indeed yielded mixtures containing RNA molecules with heterogeneous backbones or peptides with mixed chirality. In some cases, I think they should stop there. In some cases, elongated at their side chains. So this is actually interesting. I didn't know that that you could actually have spontaneous polymerization and yield and you can actually yield RNA molecules.
I knew with RNA autocatalysis was well known.
With montreal clay.
Yeah, it's not. That wasn't something that was too surprising. I actually didn't know that we got peptides though. I knew that we got individual amino acids, but
entire peptide chains.
Yeah, I mean, look at that. You look at the figure 1, they were able to get some chains. I mean, that's just a toy example, but still.
Wow. So I hope they go into detail as to how they got the polymerization to happen.
And they have, what, 4 citations backing that peptides, yeah, they have 4 different citations from, oh, look, they're all after 2002. Huh, that's interesting. It's very interesting.
But in figure 1, the RNA is linked, not the same way each step.
Yeah, this one has a 3' 5'.
That's what they say. It's mixtures. That's why they said heterogeneous backbones.
That is strange. Yeah, this one's a 3', 5', 2', 5'. Wow.
Well, because theoretically it could take any oxygen or hydroxide to use for a link, and there's both a 2' and a 3' oxygen molecule.
Are these mixed polymers functional in any way?
I don't know. Well, I think the next sentence says, elucidating the mechanisms by which functional prebiotic polymers were selected and further amplified may offer ways to resolve this prebiotic chemist's nightmare. So I think that's the question they're trying to get at, is how do you get to the functional molecules? molecules.
Hmm.
And they say achieving this aim may explain the early molecular evolution processes that led to biology as we know it today. Honestly, given how clumsily written their abstract was, this is a very good introduction so far.
Yeah. I do like how they state right in the forefront. Yeah, we've gotten this shit, and this is a pretty simple figure to kind of really introduce the topic. Actually, let me full size image. Is there a bit more? No. Okay.
But it doesn't make any difference, I think. Or
No,
maybe in the live chat. You see, in the live it's a bit smaller. Yes.
That's all right. I thought that it had an additional part of it. Let's see. In the context of the emergence of an RNA world, researchers have suggested that RNA was preceded by transient informational oligomers that were compatible with pre-biological pathways, which in turn could support the transition to RNA itself. That's interesting. What if it's transit?
What oligomers? RNA oligomers.
Yes, transient informational oligomers.
Transient.
So
yeah.
even
or just something that was informational that was pre-biological that supported the transition to RNA itself.
But not necessarily nucleotides or maybe protein-based.
Not necessarily. It could be protein-based. It could be, I think crystals have been forwarded as a possibility.
That's interesting. And there's two. Yeah. Oh, okay, that's a really recent one. On the emergence of RNA. Yeah, I was just checking the citations on this. That's interesting. Okay. Based on RNA replication experiments, it has also been proposed that the difficulty presented by chemical heterogeneity to the understanding of early evolution has been exaggerated, since some chemical heterogeneity is actually tolerable in polymerization and replication processes.
Oh. So they are functional in a way. My previous question is now answered.
Well, I think the idea is we have to get out of this mind space where everything has to be perfectly functional in the beginning.
Yeah. The creationist mindset summarized.
Yeah, that's the creationist mindset when we're dealing with what are probably imperfect structures.
Yeah, I would imagine, especially in how tumultuous the early world would have been, that you just all of a sudden have these pristine molecules that just all come together and do a perfect little dance. They all, have all the polymerization they need, and then magic happens, and then life. No, I think that you do have a bunch of effectively junk that, given the right conditions, can produce something that's conducive toward what we know today as life. And it probably went through an infinite number of steps to get there. I mean, that's what we see in nature, where you have certain species that have all types of adaptive traits. Some of them thrive, some of them don't. It's the same story over and over again, just I guess on a simpler scale. Let's see, with respect to chirality, several studies have shown that even minute differences in abundance, i.e., I can never pronounce this, enantiomeric excess, can be amplified when various self-assembly processes, including I think you mentioned it, Sam, including crystallization occur. And there's like, 1, 2, 3, 4, 5, 6, 7 fucking citations on that. Jeez. Yeah. I can never pronounce this word. Oh my God, fuck me.
I think you pronounce it beautifully.
And then enantiomeric. Yeah, that's because I look at it, I'm just like, oh my God. But yeah, that's a little pet peeve of mine. I always want to pronounce stuff correctly. And you can ask Sam about that. Anyway. I just lost it. Yeah, because I think Sam had just mentioned one of the possible ways that he had heard of, and there it is, crystallization. Sure enough.
Oh, you mean extra crystals? Like geodes?
That's crazy. Check the live chat. Where's Rational Mind? Another interesting recent paper on abiogenesis was published in the same journal, The Origin of Primordial Genome Through Spontaneous Symmetry Breaking. Huh. So I haven't wrapped my head around it yet, but I get the impression there's a lot of potential. Rational Mind, we can invite you, man, if you want to come in. Let's see. Next sentence. In addition, it has been postulated that the delivery of enantio-rich amino acids from outer space was the origin of homochiality. And I think this is what going into a bit of panspermia, if I'm not mistaken.
Well, partially.
Yeah, not adopting the hypothesis wholesale, but just I think that's a fairly non-controversial point, because we have meteor sites that are rich with certain elements that really you wouldn't see too much of on the surface, or if you did, you see it in small amounts. So that I mean, I don't think that's anything that's controversial at all.
Yeah, I remember someone asking me for how do we know these 20 amino acids could have formed without life, and I showed him a citation about a meteorite which contained over 22 amino acids, and he never got back to me about that.
Yeah, I mean, there are all type of combinations for amino acids. There's not, I mean, the ones that we commonly see for life aren't that's not an exhaustive list. There's just the ones that we commonly see. So we don't know that there or even other combinations that could have been used to create life that are just no longer suited for this environment. So
Rational Minds said that it's bedtime for him, so he won't join.
But okay, yeah. Oh, it is getting late there. Yeah, he's in the UK. Oh man, it's 4 p.m. here. It's probably damn near midnight.
Yeah, and with me also. In about 15 minutes, I must go bed too.
Okay. Active polymerization and what is it? Racemation? Is that how you say that? Racemation? What is that?
Don't get the wrong idea, I think. Don't get the wrong idea.
Racemization. Okay.
When you have a racemic mixture, it's mixed with both left-handed and right-handed molecules.
Oh, okay.
So if you were to racemize a mixture, it would mean that you have both left-handed and right-handed molecules.
Oh, wow. So this is the politically correct process here. That's what that is. Okay. So I'm just joking. Racemization. All right. So active polymerization, racemization processes and differential breakdown of incorrect products, followed by channeling resources into the correct product, compromise additional putative pathways for the formation of chiral peptide oligomers.
Oh, wait, just for your information, Rational Mind lives in Sweden.
Oh, he's Sweden?
Yeah, Sweden. He says right there. Or maybe
you're not American. You're an ISIS, bro.
I don't know why I thought he was from the UK.
He is from the UK, but he's going to grad school in Sweden.
Oh, okay.
Oh, yeah. Yes. Okay.
That makes sense now. Okay, so this is definitely above my pay grade here. Notice by my bafflement of this word here. but yeah, I think it's so differential break.
My understanding of this is basically that it's really hard to get the exact or no, what is it saying? Active polymerization, racemization, differential breakdown of incorrect products. Oh, so basically it's saying that if you break down as you're forming more products and then you break down the incorrect isomers, then you're going to have the correct isomers.
So basically, if you break down the wrong isomers and use the breakdown products for the same process, you get them back. How do you call it? A feedback loop. basically, into the right product.
Which I think is fairly well known that does happen in chemical reactions.
Well, I have a question. What happens when we eat the wrong handed isomer of proteins or something like that?
I don't think we ever really do.
I'm just curious.
I have no clue. I would guess probably not anything crazy, but I don't think we ever do, so I don't know how we would find out. Think about it, most of the sources of proteins that the entirety are from living things that are multicellular, and all multicellular organisms have the same handedness amino acids.
Yeah.
That's interesting. Let's where is? There we go. All right. Furthermore, regio. Oh fuck this word again. Okay, regio enantioselective. Excuse me. In the steps of chain elongation by growing beta sheet peptides yield isotactic sequences, albeit often forming racemic aggregates. Oh.
So the million dollar question: what the hell does that mean?
Regio enantioselective.
I think it's saying that it's selecting a certain enantiomer.
Oh, okay.
I don't know what an isotactic sequence is, though. I've not heard that terminology. This is really stretching into like how much orgo and mol bio do I remember? And it's clearly not enough to understand this paper fully.
Okay, isotactic polymers are composed of isotactic macromolecules. Okay, well, in isotactic macromolecules, all the
I see. I see me from the picture. Yes. Yeah.
All the substituents are located on the same side of the macromolecule backbone. Huh.
Yeah.
Yeah, this is
it's pretty self-explanatory from seeing the 3D pictures. That one and the one below.
Yeah, you can see the orientation of where the. So I guess these are what functional groups out here. I don't know. Actually, no.
The R's are functional groups.
Yeah, the R's are okay, and so they're okay. So yeah, their conformation is on a particular side, and that's why they're called, I guess, isotactic.
And which is necessary for to have a template, a functional template.
Yeah, because
I think.
It makes sense, because if they weren't in a particular configuration, then they wouldn't be able to form those weak bonds that allow them to what's the rational mind saying? Denoting a polymer in which the repeated units have the same stereochemical configuration. Okay, so they're uniform.
Yeah, it's essentially what we were saying.
Yeah. Where did I leave off? Okay. Interestingly, a recent study with RNA has shown that both cross-catalytic and anti-cross-catalytic of one-handedness or the other are tolerable.
What does it mean by cross and anti-cross catalytic?
That's a good question. Boop. Put it in Google.
I don't know what anti-cross catalytic means. Again, this is where my limited level of biochem is showing.
Cross catalytic replication. Something to do with ribozymes.
Yeah, I'll just put this in. It looks like it's something you can't just look up. Oh.
No, it's the technical term that only is used in papers.
Yeah, which does happen, folks. So it's a little bit of homework to do. Actually, you know what I have? I have a fucking Doctor. I'll just ask Dr. Rio tomorrow. I think I go to him tomorrow, so I'll ask him. Let me copy that, and I'll send that to myself, and I'll talk to him about it. Because that's what we do, folks. If you don't know something, you know somebody who is in the fucking field, ask them. Hold on, give me just Just a second. What on Facebook? Oh God, holy spackle. God. Okay, so let's get.
What did the Holy Spackle say?
She put a quote from Shani in my inbox.
What was the quote?
Hold on. That's not it. I was just sending you what I'm going to send to my instructor. The quote is, "The Spirit of God came in my, and it's kind of hard to contain him."
Ugh.
Oh. So there's a nasty image.
Can we continue?
That sounds not gay.
Oh God. Okay. Herein we put forward another selection mechanism relevant to early chemical evolution, since we probed the instrumental significance of non-enzymatic replication for the amplification and fixation of certain products within a heterogeneous prebiotic mixtures. So
they were putting forth a suggestion as to how it could have tended towards that handedness.
Without the without the use of enzymes and how it became fixed. See, in particular, we demonstrate that beta sheet driven peptide replication can be used for significant enrichment of native condensation product in previous or preference. To some of its regioisomers and stereoisomers, and henceforth correction of the evolutionary drift towards non-functional heterogeneous mixtures.
So basically, the templated replication can explain why the heterogeneous mixtures are non-functional and why it doesn't drift towards that. Pretty cool.
Functional. Let me see. Copy this before I read it. And the involvement of peptides in early evolution has been advocated several times. Notice all the citations, and has also been suggested as a complementary or alternative route to the RNA world, leading to primitive life-like functions, and a shit ton more citations. Keep note, folks, all these citations for these, like this is, you know, that thing where some of the CCP members will go, hey, you know, that's a positive claim. Where's your evidence? There's literally the citations here right after each claim. So if they have any questions, the link is down there. That's after 2002. That's after 2002. All of these seem to be after the shit that, well, except that last one. That last one's like 1996, I think. Yeah. So recent studies have indeed revealed multiple assembly pathways yielding amphiphylitic peptide fibrils and nanotubes, and consequently the emergence of new functions such as catalysis, binding of nucleic acids, and charge transfer.
So they are also very functional, very indeed.
Yeah, I mean, if they can do all this, then they're on their way to being what we need them for as far as life is concerned. Where is this? Of particular relevance to studying the origin of life is the search for oligopeptides spontaneously formed and evolved as autocatalysts. I think this is important because it's getting over that issue of needing certain proteins to act as enzymes for anabolic and catabolic functions. So if you have certain molecules that can, you know,
I think if you just say catalysis, because not all reactions are either anabolic or catabolic.
Oh, okay. So just say what now?
Just say catalysis, like catalyzing reactions. I don't think you need to mention anabolic or catabolic. It's not relevant here.
Catalysis. Okay. Note that. Accordingly, we have previously studied the replication of a simple amphiphylitic peptide, and that's in one figure 2A, that when forming beta sheet architectures can efficiently accelerate its own formation from a solution containing the appropriate precursors. And where is figure 2A? See if I can, I can't make it bigger. I wanted to show folks. So yeah, what they're talking about, folks. If you click on 2A, which okay, oh here it is. Yeah, this is what they're talking about here. Go back.
So these sequences were spontaneously formed, and they showed that they do this thing, self-replication.
Yeah, and this says that they have the right precursors to what you, I guess, expect from early life, or even, I guess, pre-early life in this case. The current study takes a different direction in that we applied original design and experimental conditions for inducing spontaneous evolution of a mixture containing isomeric compounds, each presenting a different phenotype following self-assembly. It's evolution of mixture.
Dangerous words.
Use the word phenotype. Oh shit. Where's the next part?
The tongue will come in and say and scream epigenetics.
No, don't. God, don't say his name. We thoroughly analyzed the complex web of reactions in this network via experiments and simulations with the aim to elucidate the role of the initial conditions, seeded precursors, the stability of the intermediate compounds, yielding different products, and template-assisted replication. So, and I would imagine running many simulations for the scenarios that we had mentioned before, where you had the breakdowns of the ones that didn't work, and then the fixation of the ones that did, to see, how viable is this scenario. So that's where, again, we have the use of computer simulations that we didn't have 20 years ago to help us with this kind of stuff. And it actually speaks here the seeded precursors that help facilitate this process. So I guess once they determine, what the intermediate compounds are, you can then see, well, how they branch off from there, where, whatever compound X can either go to Y or Z. Where was I? There we go. Helps when I highlight shit like this. I don't lose my place.
Two components.
Yeah, there. Oh, geez. Okay. Remarkably, we find that when the replicating molecule is also the condensation kinetic product, modest replication exerted, but when the network reaction is initiated, the materials leading primarily to other products with a non-homogeneous backbone, efficient correction takes place by replication and enrichment of the native peptide at the expense of other network components. So the condensation kinetic product. Are you familiar with that, Neslik?
No, I don't think so.
Proteins are formed via condensation reaction.
Condensation kinetic. Oh, okay. I see what you. Yeah, there we go.
Just saying that the product of the condensation reaction.
Okay.
Oh yeah. No. So I think we use a different word in our language to describe condensation. But now I get it. It's a reaction that takes water molecule from
you have the functional group of a hydroxide, and then you combine that with loose oxygen or something, and then it'll precipitate a water out, so that the other so that those two can combine. Yeah.
Yes. Now I remember. Yeah.
Yes. You mean a proton, not a loose oxygen.
Oh yeah.
Oh, did I say oxygen? My bad. My mistake, folks.
Hydroxide. Damn it. It's a proton, and was it the group called
hydroxide? Yeah, I said oxygen. I said hydroxide and oxygen instead of hydroxide and proton.
You get HO2.
So basically, it's saying that there's replication at each step that's at the expense of other components in the network of products, which basically means that preferentially some orientation is being being produced, which would explain why there's an affinity for certain isomers and not others. Pretty cool.
Yeah.
We conclude this paper with a brief discussion of some implications of our findings, highlighting plausible relevant pathways in early molecular evolution. Okay. And did it mention this? Because okay, no, it did figure 2A, but doesn't mention 2B yet and 2C. Oh, okay, no, this is where they mention it, looks like. So here's some graphics, folks, as peptides utilized for the current study. And let's see, A, peptide name and their sequences. There's A. Okay, yeah, that's what that corresponds to. So you guys can look over this because it's explaining what these 3 graphics are, or figures, I should say, not graphics. So results. Spontaneous evolution of a peptide network via glutamic acid ligation. This study focuses on the unique non-enzymatic replication that takes place in a small network made up of peptide 2 and its isomers, and that's also in figure 2 that we just looked at. The sequence of this peptide contains only repeating, was that glutamate? That's not phenylalanine, is it?
No, I don't think so.
PHE.
I don't remember the abbreviations for the amino acids too much.
Oh, that's going to
I think it's glutamic acid.
Yeah, I know this is glutamic acid. Yeah. PHE is phenylalanine.
Yeah, I'm pretty sure it's phenylalanine. I can't think of another one that's PHE.
Yeah, I think, yeah, so glutamic acid, phenylalanine residue, dyads, and caffeine groups. Yeah, still referring to the other graphic. It is accordingly simpler than peptide 1 and potentially more prebiotically relevant. Huh. It looks like, I guess there's somewhat of an affinity for this. Oh yeah, because I see the glutamic acid here, and they've highlighted it here.
Yeah, I look it up. It's phenylalanine.
It is? Okay, yeah, I thought it was. I'm surprised.
I can't think of what else it would be like.
Yeah, there's only so many, you know, amino acids that start with a P, but then PH like that. Yeah, that's. Yeah.
Well, it is weird. I should have remembered that one because I worked with that amino acids on a project during my college.
Oh.
I had to memorize.
There was one. There is one bacteria that converts phenylalanine into a compound that resembles the rest group of that amino acid, and I don't remember that name. I think toluene, something like toluene.
It's been a while. It's been a while since I've looked at a list of amino acids. I just so happen to remember phenylalanine because it was. I think we might have been using it for an experiment. We're talking about it for an experiment. I don't remember. Anyway,
I put the compound that here in the side chat or in the group chat. Toluene. That's basically the compound that is attached to this amino acid, the phenylalanine. So basically, it is the bacteria that cuts this group off, and you have like a very useful compound. But moving on, not about my project. Yeah.
Where's it? Oh, there it is. Okay. The main differences between the replication of 2 and the observed earlier for 1 originate from the mechanism of their precursors ligation. Yeah, this is referring back to the graphics. I think all this actually section is referring back to what's happening with the graphics. Wish I could blow this up a little bit bigger so folks could kind of see, or if we had an animation, that'd be even better. The replication of 1 was driven by the efficient and selective native chemical ligation reaction propelled by an alanine thioester electrophile and a dedicated was that cytosine residue cysteine residue at the nucleophile N-terminus.
Oh, it's already 7 minutes past my time. I should go now. So thank you guys for having me, and see you next time. Bye.
All right, see you, man.
See ya.
Okay. Yeah, so you said that's cysteine? Yeah, CYL, yeah. I don't know why I thought there was a T there. Yeah, actually read that again. The replication of one was driven by the efficient and selective native chemical ligation reaction propelled by an alanine thioester, or actually, when I say thioester electrophile, and a dedicated cysteine residue at the nucleophile N-terminus.
I'm not familiar with the specific reaction, so I can't break that down. But basically, they're saying it's self-catalyzing, the ligation.
Oh, okay. So I get, yeah.
I actually don't know if it's self-catalyzing. Driven by the efficient.
I thought that was somebody knocking on your door. Yeah, I was trying to find. I'm trying to find a little bit more background information on that, folks. Yeah, probably could have somebody explain this a little bit better. Yeah, Lexi, you know what? Yeah, I'm going to copy that so I can look some more into it later.
Sorry, we have painters at the apartment complex today, so I have to be. They asked for one person to be home.
Ah, gotcha.
But from what I can tell from looking at that, so you're gonna have to bear with the annoying knocking. Sorry. Is that they're referring to the reaction in reference 53, and so that's what's driving the replication of protein of peptide 1.
Let's see. Synthesis of protein by native chemical ligation. Okay. Oh, yeah. So, yeah, if folks are curious about that.
There is zero chance that I'm going to be able to understand that paper. We are right now at just the limits of my biochemistry knowledge.
Yeah, mine was expended early on because, yeah, this stuff gets into it, folks. This, long story short, for the rest of this, it goes over, like you see here, you can see the figures of, looks like, what is that?
Well, do they have a figure that, I'm looking at the paper, do they have a figure that, okay, so I think figure 3 is the replication of these mixtures. And it looks like 2 is replicating. Yeah, this is beyond me.
It's figure 3A. Is that spectrometry? Because I see that they have what that the gamma nanometers.
No, that's it's wavelength. Yeah. Structural characterization. Yeah, it's a spectra.
Spectra. Okay.
But this is beyond what I'm capable of analyzing. And then they actually, in figure 5, they actually have the chemical reaction.
5. Yep, there it is. Oh, okay.
So yeah, we're way beyond what I'm capable of.
But
I can explain what's going on there. But yeah, I think we've hit that point of the paper where I am not going to be able to help people understand this any more than I do.
Yeah, long story short, folks,
papers are hard. Yeah,
it is. You actually have to sit down. Like when I first read through or tried to read through Sam's thesis. I couldn't. Like, I had to stop, and there was a limit to what I could discern from like anything, just like basic stuff, you know. But it's important to know, and I wish I had that image because Jade put it up earlier. I say earlier this week, where you have movie scientists, and it's like a movie biologist, and they're looking at something like a computer monitor, and it has something to do with physics. And then the movie scientist goes, "Oh yeah, that's no problem. I got this." He's just cracking his knuckles, but the actual scientist goes, "Huh, I might need some help on this." And so he's eliciting the help of somebody like who is a physicist, and that's why you have all these specialized fields. And so overall, you can learn as much as you want about your topic and know a little bit outside of that, because we all have some, well, I say all, people who study science in one field know a little bit about the others, but there's just a certain point. There is that gap where you go, all right, I'm going to have to pull in somebody who has actually taken, you know, analytical chemistry and, you know, techniques 1 and 2, where they know specifically what's going on here.
I don't even know if that, I mean, I think to understand this, you would need really, really upper level biochemistry, which I just don't. I'm looking through the figures right now. Maybe we should just skip to the discussion, because my ability, I've fully confessed, my ability to understand what these figures correspond to is severely limited.
Yeah, and one of the things that I just wanted to stress for folks is that, yeah, there's no shame saying, you know what, this is my limit. This is, I don't have to pretend to know more than I really do, or get on the internet and pretend to know more than I really do. Sam didn't have to pretend to know more than he really does, unless he's trying to impress a girl. Then that's different. That's very, very different.
No, I know plenty. I don't need to. I don't need that. I'm good.
Yeah, but so yeah, if it is important, like if you do want to reference a paper at any point in time, you do have to understand at least a lot of, I would say, not just read the abstract, because that's, I don't want folks walking away thinking you just read the abstract and that's it. Nephilim Free does that. He doesn't actually understand contents of a paper, and he gets shit wrong. And there's several others. Tommy Hall does that too. At least if you go through the discussion and you see what's said and read everything and not just the first few lines or what you want to get out of the paper, oftentimes, and I've seen this in a few papers, there'll be caveats. And it just depends on that person's writing style, which whoever wrote this paper, they have a very interesting, I'll say that, writing style.
They've got to be foreign, because some of their sentences make no fucking sense. Like they're trying to, they're cramming so many prepositions and verbs.
I don't know if I'm tired of seeing commas or what, but
they're from Israel. Yeah, they're from Israel. Okay.
Because I was like, some of that shit could have been broken up and written a lot better than that. Yeah. But yeah,
they're Israeli. But do we want to read the discussion? Because I'm not in a position where I'm familiar with these figures.
Yeah, no, yeah, we can definitely go to that. How you doing, dear?
I'm looking at these figures and I have no clue.
If you want to go ahead and read that, the wife just came home and I need to chat with her for a little bit.
Alrighty. Okay. The common practice of sequence-specific oligomer synthesis in organic chemistry makes use of very selective coupling reactions and the incorporation of protecting groups into reactive side chains. DNA polymerization in biology is, however, controlled by a different mechanism, namely post-replication error correction, preceding by removal of misincorporated units and their replacement for the correct ones. So they're talking about the difference between the type of reaction that they're doing here, where you're using a templated reaction versus having the error correction machinery in our cells. Any questions about that, or is that meaning... Okay.
All right.
Both the synthetic and biological strategies were obviously not applicable to polymer synthesis on early Earth. Scientists were thus looking for other universal mechanisms that could support sequence-specific oligomer synthesis that were accordingly attracted by the idea of post-synthesis correction mechanisms. Nevertheless, in the absence of efficient catalysis by nucleases or peptidases, the reversible processes needed for breaking the nucleic acid or peptide backbone and for recycling their fragments as building blocks in correct synthesis are very slow.
God, that's horrible.
It's a very dense sentence. The one starting with nevertheless, that's a very dense sentence.
It is. That could have been, okay. God. For that matter, dynamic covalent chemistry was developed recently, enabling continuous equilibration of building blocks and products, thus leading to selection of the most fit compounds in the mixture, depending on the applied environmental conditions, templating molecules, and sometimes even function.
Huh. That's interesting.
This is the name of the technique, dynamic covalent chemistry. Huh.
Yeah, I don't know what that is. Like, this is the problem, is I'm not a biochemist.
Yeah, they said they just developed it. So shit, this actually highlights what we were talking about, why you need recent stuff. If they just developed this and this is something that is on the cutting edge, then this is more relevant than something from 1992. Way more relevant. Let me see. However, as of now, DCC cannot support reversible chemistry of native peptide bonds and is consequently only partially useful for studying prebiotic protein evolution. Well, I'd imagine that they can't simply put in, one of our proteins and just go, all right, magic, and then hit a button.
Well, I think the idea is that a lot of this protein chemistry involves reversible reactions.
Oh, yeah. See, I took that too literally.
Well, I thought I took it pretty literally, but I don't know.
As in reverse. No, when I say reverse, when I say I took it literally, I was thinking reverse engineering them.
So you took it literally in the sense that you read the wrong words.
Exactly, you got it.
So Ronnie's version of literal.
Literal, where it's not really literal, it's something completely different. But look, folks, if you listen to 5 minutes before what I said and then listen to 6 minutes after what I'm saying now, everything will make sense. I promise.
Yeah.
Or I just thought the wrong fucking thing. It's really easy just to go, you know what? I just read that completely wrong. It's all right. It's not a big deal.
There's nothing about beaks that would imply that they turn into ATP synthase.
It's not wrong, is it? Or is it?
No, it's definitely not wrong. It's such a dumb claim to be right about.
You can't. It's not wrong. we have Go ahead.
We have consequently searched for another systems chemistry approach whereby the enrichment of one master sequence is a result of a collective network behavior manifested by multiple template-assisted processes. That's cool. That's pretty cool.
Master sequence. So like the progenitor sequence? Like, is that?
I think the most fit sequence is the result of multiple template-assisted reactions.
Trial and error.
I think that's what they're saying, which is fucking cool.
One sequence to rule them all,
I guess.
I had to sneak in a Lord of the Rings reference.
Yeah, I mean, it was asking for it.
Right. Let's see.
I mean, it's better than me doing like a Master Race reference.
Yeah. Oh, yeah, because that definitely worked out well last time, huh?
That would have been a little much.
Yeah. Why do I owe $50 to them? What's going on? Tell us this. Okay. Anyway, similar to previous reported results with coiled coil peptide. Okay, that's redundant. Coiled coil peptide networks. the synthesis of native or wild-type peptides in our system is significantly enhanced in a small isomer population. So what a particular type of peptides, the coiled peptides within the network, and it was like what able to make the native or the wild-type peptides.
Significantly enhanced in a small. No, it's when you have, well, when they first were doing coiled peptide networks, because coiled is it's a type of chemical orientation of peptides. This is just saying similar to that. It's that you know, the synthesis of a new peptide is enhanced when there's a small isomer population. It's only when you have only a few isomers you're able to have a good ability to produce a wild type peptide, which actually makes sense for abiogenesis, because when you think about it, there's not going to be many wild type peptides out there. There's not going to be many isomers because this is a fairly rare process. I think that all kind of fits in.
Yeah.
Or I could be talking shit. This is definitely not my field. So
that was a 50-50 chance, folks.
50-50 chance at best here.
So our results hint at an alternative backbone correction mechanism that could have been developed within simple chemical networks through the emergence of shorter beta sheet polymers. So you almost got me saying beta sheet polymers with efficient and selective catalytic behavior. I caught myself before I said it. Oh man, okay. So an alternative backbone correction mechanism. So yeah, minus, you know, enzymes that can do shit, you know, including catalysis. This is an alternative, and it could have emerged through just smaller or shorter, rather, beta sheets that we talked about before, and worked effectively within that environment. That's what it sounds like.
That's what it looks like. Yeah, that's pretty cool. In such a scenario, the formation of several products in a mixture is not critically harmful, since this is accompanied by slow progression of the production of one molecule at the expense of others, leading finally to the formation of dominant functional polymer backbone. Okay, that makes complete sense to me. We do have the correct interpretation, I think.
Okay, good. Yeah, because I think we talked about this earlier, where, you know, the incorrect, we'll say that, I guess the incorrect combination of molecules would not have lasted in that environment. So they'll been, I guess, effectively recycled for the more favorable ones. So let's see, there it is. Okay, in the future, we wish to study the relevance of our approach to replication systems, facilitating the condensation of more than 2 peptide units, and ultimately even the sequence of specific addition of amino acids. So while they have the production of particular peptide bonds through condensation, seeing how viable it is when you start adding more to those chains, because, you know, we have large macromolecules made of tons and tons of chains, tons and tons of peptide chains.
And I think they want to scale this up, right?
Yeah,
that's so what would have to happen in biology.
So rather than saying, "Oh, this looks good, folks," you know, this is it, we found it, you continuously check and challenge your own thesis, because as the chains grow, they'll begin to get their own my dear child, their own structure, their own physical structure. And that's why we were mentioning the conformations, or I guess the arrangements of the beta sheets. You know, we spoke before about what happens when you have large chains of molecules that aren't properly folded. They can't do their job, and sometimes they destroy others. So naturally, the success in developing such system will depend on further improvement in selectivity toward the desired sequence. Again, if it's shown that, hey, the ones that we're looking for are proliferating, then, well, shit, that's a good thing. Because if they're not, then, I mean, it's kind of back to the drawing board, because you expect the ones, or the desired ones, to be the ones that are beating out all the others in the given environment. Okay. Finally, we note that our results are in line within the old notion that the emergence of primitive replicator was crucial and potentially sufficient for the origin of life further down the road.
Well, I think that's a good ending.
Yeah, it was. I mean, it wraps everything up. So, all in all, folks, hey, it does take a little bit of know-how to read this stuff, and even then there's still limits. So feel free to look over this, as sometimes you just can't Google what certain things are. Sometimes you need a bit of formal education to really dig into some of this stuff. So as autodidactic as people think that they are, there are
Well, you
certain
also need
limits.
the correct education too.
Yeah,
Like, I have
God.
plenty of formal education, but this is way out of my research area.
Yeah, you can't just pick up any random, I don't know, nursing book and hope to learn this stuff in a weekend.
But I think that's just about it.
Yeah, it is. Hopefully, folks, y'all may have learned something. That's why I tried to go a little bit into the science that we have done. I'm in chemistry now. I was actually doing chemistry homework before I was doing this. So, if anybody has any questions or they find some papers that I guess are more relevant to our fields, that'll be a bit better. But yeah, this is
Well, I can
definitely.
present that paper that I was talking about. That was the basis for a lot of my research currently.
That's not the one that had your name on it, was it? Or was
It does not have my name on it.
So there was something you were showing me that also had your name. No, that was something else. No, you see, that was the basis for the work that you're doing?
Yeah. I mean, granted, I know the scientists who did it, but...
Okay, that might have been what it was.
But that's different. It's a small enough world. Like, I also know Manolis Kellis. I've met him a couple times. That doesn't mean I know him, or interact with him.
Well, G-Man and Ronnie can go up to him. Hey, you know this guy named Sam the Scientist? He's a scientist. You may have heard of him. When Doc drop you.
Yeah, I don't think Manolis Kellis would know who the fuck I am. We've met once. He's at MIT. I don't go to MIT.
Questionary structures. You need it. Yeah, and RSC. Yeah, you are correct. Yeah, the tertiary and quaternary structures. You need chemistry background to understand this. Don't tell lay people to go and read. I want people to go and read on their own for a reason. You can learn some of the basics on your own. I don't want to say that, well, you need formal education to understand a topic, but the higher up you go as far as the difficulty of the topic, you will need some type of help with that, and is best found in formal education. I don't know anyone who's become a forensic scientist or a fucking chemist by reading random nursing books, unfortunately.
I also don't think that's a fair accusation to lay at either of us, right? Like, we've both said on multiple occasions people need to learn the basics. We're not the ones who think. There are a lot of people in our community to quote a brilliant president, on both sides. No, but there are a lot of people in the great debate community, regardless of theological conviction, who think that they can just jump into science when they have, scientific publications, when they have a high school level understanding at best of these things. And I've said this over and over again. So I don't think that's necessarily a fair criticism, at least not of me. I think you've said similar things.
Yeah, I have. I want to encourage people, outside of school to do their own studying, but also understand their limitations and understand that, hey, know those limitations, recognize them, and go, how can I surpass them? And then that's really it. I don't expect anybody to just jump into this paper and try to use it in a conversation. You can defer to it and say, you know what, there are papers out there on this topic that are way beyond you and I both. So I know the research is there, and that's about it. Like, you can give the caveat that I don't have the knowledge, I don't have the requisite knowledge, to truly understand it. So If you're saying that the research doesn't exist, then no, that's false.
Fucked. Yeah. And also, yes, RSC, sorry. I was quoting Trump.
Oh, man.
But I think we've pretty much got it covered.
Yeah. Like I said, we were going to give a commentary about that anyway, because there has been a lot of talk, and of course I watch almost everything that people send me as far as what G-Man saying about formal education. It's like, yeah, I'm sorry that I'm not sorry that I do value formal education after not being able to go to school for X amount of years and knowing what I wanted to do. I value it. That doesn't mean that I just sat and languished while I was waiting. No, I educated myself and understood that I was going to reach a certain place where I couldn't progress until I formally attended some type of university or college or whatever.
Well, there's no effort on the hands of some other people. I mean, but there is a good point. It's like you can't get angry at people for pointing out that you don't know what you're fucking talking about, and a lot of people do that. Like I was the first person to say, maybe me personally, I don't understand what's going on in this paper, right? Like I've hit my limit. I didn't try to understand something I just fundamentally didn't have the foundation to understand. And I'm sure that, if I spent hours upon hours trying to scour through it, but practically I don't have the time to do that.
Yeah, it's just not really feasible given, well, I know what you do, but given what, we both do, there is a limited amount of time. And yes, we have dedicated an hour to do this. You can relegate time, but some of these tasks take more than an hour of just sitting down and talking about to really perform, effectively. I'll say that. So on that note, anything in the live chat, folks?
I guess we can open it up.
Let me just put it out there.
There's a certain person in the GDC by the name of Brian Stevens, the one with the mad scientist avatar.
Oh, I've seen
gets angry when you point out that what he says is so much of the time even more incoherent than what G-Man and Ronnie say.
I've seen his avatar, but I haven't actually.
Don't read his posts. It's a waste of life.
I'm assuming you're not a big fan of his.
Well, he just puts shit forward where it doesn't make sense. And then he gets angry when he's like, I'm sorry, I'm not good at communicating. I'm like, don't get angry at me. That's your fault. Like, I don't know.
You're just being mean. You're just being mean, that's all. Improvement in selectivity toward the desired sequence. So cytosine. No, I don't think it was cytosine. No, I misspoke.
It was cysteine.
Yeah.
We're not talking about DNA.
Yeah, when I said cytosine, I misspoke. It's definitely cysteine. But
we're not talking about DNA, though. We're talking about protein. So anything about cytosine is not going to be applicable here.
Right. All right. Links out there, folks. Let me go check. Let me go check G+. Send me messages, and I tend to forget to look at it.
I'm going to switch to my phone. Okay.
Okay. What's this? What is this? Oh no, I was trying to see if anybody sent me a message, and I saw there was the Hangout invite group or the invite list, and there's a bunch of people. Oh, I got notifications there. Steve said Blue is on a long timeout. So
what happened?
Let me see.
Blue's really angry at me right now, and I don't fucking know why. Like, he's angry that, like, apparently I can get into the live chat of Hangouts while code is running. I don't know why that's really... Is it that crazy? If I have shit running for minutes at a time and I'm listening to it while I'm coding so I can pop in... You've seen I've run my programs in front of you, so you can validate I'm not lying.
Yeah, there's nothing you can do when, fucking over 600% of the processors are being used to run code that's going to take for fucking ever. You can't tell it to go faster.
Yeah, I've sent you when we had our server running at like what capacity was it? Like 99% capacity between me and Chris.
Yeah, like, that was ridiculous. I was like, how many cores is that again? Fuck.
56 cores. 56 cores.
Damn. So, needless to say, folks, you can't force it to go any faster at all. It's just, so you will have a little downtime, just a little bit. I'm trying to see what Maynard's putting. I'm confused.
I don't want to call it going full true, but you're just jargon bombing now.
Yeah, I'm not sure, but because this isn't about DNA. This is simply about
peptides.
This peptide. So we're talking about like the bonds between amino acids forming that I guess you could call it the precursors to proteins, but like you can't. There's no DNA involved in here.
No, it's not relevant to the paper.
I put the link out there if anybody wants to come and chat. Heavy, if you want to come and ask some questions.
I don't know how equipped we will be to answer those questions, but we can try.
I mean, if they're like basic chemistry or chem, yeah, sure.
Yeah. Beyond that, you're getting into testy territory here.
It's like, good luck, everybody.
What did TE say? He said so many things about DNA.
Have your pick. Like, what do you?
Why is Blue in timeout? That's hilarious.
I think he was getting in an argument with somebody. Hold on. Blue is on a long timeout. He is blocked by me and Redline, just FYI. And Steve just came in and said that. So let me scroll up. Oh shit, was he arguing with a bunch of folks in here?
How do you unblock people? Because I don't know if I'll be able to see his. What is this post?
It's in
to me on Facebook.
It's in the where we send each other the Hangout links and stuff in the Messenger and the Google Mail.
Yeah.
Yeah, there's a Hangout invite list group specifically like for the GDC, and it's in there.
Which one?
Let me see if you're a part of it.
Can you just add me to the call or?
Yeah.
Or just ping me.
Yeah, I just added you to it. So because I saw him arguing in there last night with a bunch of people, including like Jackie. So I was wondering like, what the fuck's going on? But I have it on silent, so I didn't keep ringing. So evidently something happened.
Apparently he's calling Red's rhetoric a troll.
Really?
He said he is apparently really saying that feel free to fall down a flight of steps isn't a threat, which I guess technically it's not. But
well, sure. I mean, I could say that to presidential candidate too, and
yeah.
I don't think that would go with the Secret Service very well. But hey, that's just me.
Well, I wonder how long it'll be before True presents a mangled version of this for his side.
I mean, he is more than welcome to come and read this fucking paper, because I know I stumbled over a few words my damn self that I hadn't run into ever. But I welcome, like I please do, because I think it'd be entertaining.
I was trying to keep up because a lot of it went right over my head, but it almost sounded like maybe way back there was like certain combinations of chemicals or processes that kind of got the ball rolling for RNA or what makes self-replicating, whatever you want to call it. I'm stumbling here. But whatever the processes and reactions that happened back then, they don't happen now. We don't see examples of them because the conditions back then were so different from what we have now. Did that make any sense at all?
To some degree. I mean, it is true. The paradox of studying this is that life is good at adapting to situations. And, well, if we're studying where, you know, where there was no life, well, that's not going to happen, because once there's life, it's going to adapt and live there. It's going to live at any place where it's livable. That's a known paradox. And
whenever those chemicals or reactions were, they made that framework or backbone, whatever the word you was using. There's like, here's this scaffolding for all this stuff to happen, and then it starts happening, and it allows other processes to keep going. And the original ones that started, they just kind of go, and what you have left is stuff that's like replicating or something. They were making amino acids and things, peptides.
Maynard is still arguing in the chat. Does he realize that peptides have sequences too?
I mean, they kind of. Wait, what? I'm not even looking at that. Hold on, let me see.
Oh, apparently Tyler now wants to agitate blue. That seems to be the flavor du jour.
Oh God, don't do it! Don't do it! We already got one hurricane to deal with.
Hurricane blue. Hurricane blue. What set him off? What was he doing?
I don't even know. Like, because I saw it last night. I'm like, why is Jackie a part of this? And so because he was making a comparison between people upsetting him and compared to Jackie. And I'm like, I'm not getting into that. I could say something. No, I'm not getting into it. Not doing it.
I don't know why he seems to have this weird possessiveness about being one of the first people, like being like an original in this community or something.
What have we said before about like the environment? Like, this is really conducive for social misfits.
This is true.
Okay, I'm trying to install a whiteboard so my wife and I can help keep track of things. But yeah, heavy man. Trust me, man, if you didn't understand this, you are not the only one. There's a lot contained within this particular paper, and if you like, it'll be kind of hard to follow if you've never had a chemistry course or recently, because it is just chock full of stuff that. If you don't understand how chemical interactions work, just on a basic level, it's going to be really hard to follow. So that's why we try to open it up and try to explain some of that stuff to folks, because it's like I completely misread the shit as far as the condensation reaction, and I misspoke when I said it was like a hydroxide and an oxygen, when it's actually a hydroxide and a proton. So I guess an example. So you would normally, like, if you wanted to form a chain, like think of like a chain link fence, and you want to connect one chain link fence to another, you would find something to bind it, like a wire, right? And you're like, in our world, we'd find something to tie it with. In the realm of chemicals, imagine that you took part of where you're going to make that bind at, and you then just like tore off a piece of wherever you want to try and bind it, put the two pieces together you want to bind, and then use that piece you tore off to kind of you just take that part off, but then that part,
I don't know where you're going with this one.
Yeah, I thought
I'm bailing out on this analogy right here.
You could take it and you weld it in, everything's cool. No, I was like, no, that won't work, because that has nothing else to do with it. So okay, so you ever seen a video game, right? and you got this weird boss character, and you rip off part of his fucking arm, and then he combines with another boss, and it makes a fucking super boss. There you go. That part that she ripped off is no longer a part of the boss. It's its own thing. There we go.
Yeah, so it's like certain things that don't fit together. There were some chemicals or enzymes or something that allowed these things to fit together and work.
Well, you can't put them together because they're being blocked, and so that when
there are no enzymes here. This entire paper is a non-enzymatic. There are no enzymes in abiogenesis because, well, there are no enzymes. There's no proteins. We're talking about the origin of proteins.
So it's peptides, whatever those are.
Peptides are chains of amino acids.
Oh.
So you can think of amino acids like Legos, and then you like people take Legos and they make castles and stuff like that. But if you have a pile of them on the floor, they do nothing.
Yeah, that's a good analogy.
Yeah, so yeah, it's better than what I was trying to go with because that was terrible. But
I'm making a pasta sauce using the recipe from Christopher Maudy, so you know it's.
I was about to say, I hear you doing something in there. It's in the kitchen, and if it involves anything rigorous, it's from Maudy. He actually made a video for me a while, like years ago, making what is it? Chicken gnocchi. Like
what?
Chicken
gnocchi.
Gnocchi, is that how you say it?
In gnocchi, yeah.
Yeah. He actually made a homemade version.
How did you say it originally?
Gnocchi. I don't do Italians at all.
Well, at least you didn't get angry and accuse us of making up the pronunciation or sharpshooting or hiding under the bed or whatever Ronnie wants to copy from us now.
Oh man, I'm mad that G-Man took my. Have you heard him say meat and potatoes?
Yes.
Who do you think he got that from?
I should have suspected.
Nobody else says meat and potatoes but me. I don't think I've heard anybody else say it. I was like, come on. I got that from. I think I got that from my my old company commander back in high school.
Well, it's a basic saying. It's like, oh, the meat and potatoes. It's like the it's just the backbone of what you're, it's the basics. Like if you say someone's a meat and potatoes boxer, it means they use simple tools. They're not very tricky. It's not a negative thing. It's just meat and potatoes is delicious.
Yeah.
I have a pot roast cooking at the.
You muted yourself. No. But yeah, Heavy. If you can think of the building blocks as like Legos, and you put you can connect Legos together, and they can make all types of structures. Well, I need to get my hammer. Disconnected. Oh, there he is. When you connect Legos together, you don't lose anything from the Legos. Like the Legos don't get smaller when you connect them together. When you connect amino acids together to make chains of them, you'll actually lose a bit of them, and you'll lose because you can't. How can I say? So you've seen the top of a Lego. You've seen the little dots on there that you can click them or connect them with. If those edges were smooth like the sides of them were, you couldn't connect them. Okay, so that's kind of what you're doing when you're connecting amino acids, is that you're removing in this sense that where it would be impossible for them to connect to, and there's different reasons why you can't, because there's different what's called functional groups that will kind of block a possible connection, and so they'll just be
I don't know if that's correct.
Is that accurate? Or
well, functional group is the part of the amino acid that's catalytically active. It doesn't block any of the bonding between two adjacent amino acids.
I thought the hydroxide was one.
No, hydroxide's not a functional group.
Oh,
not to my knowledge. Well, maybe there is one. Maybe there.
I was thinking of
replace that.
I was thinking of carboxyl group. That's why.
Yeah, carboxyl and functional are very different things. Not to say, at least in the context of what we're talking about there.
Yeah, what's odd though is when y'all was reading over and discussing it, it was like I could kind of build up an image in my head or what you're talking about, and I'm like, yeah, that makes sense. But when I try to express it, forget it.
Yeah, it's complex.
Oh yeah.
And specified.
I think
Sorry.
it was
and
almost
sequential.
Like, oh, sorry.
No, I'm just saying. I'm just doing Ronnie's. Yeah.
Well, it's almost like it seemed like I was getting the impression that however it started, the abiogenesis process or whatever, it may not have been as complex as we think it was. Like, you're talking about like some like some chemicals or something. It's like you get some chemicals together, and all of a sudden you've got all this stuff coming up out of it, where they're combining and everything, like self-assembly. I think that's the word.
Well, it does seem like it's. I mean, yeah, it's self-assembly, and it is relatively simple beginnings. But I think that it's like very. That doesn't mean that the explanation is simple, though. Unfortunately.
No, yeah, and they've even kind of hinted that just because it works on this like minuscule scale, that they're going to have to try and make sure that everything follows suit once they start scaling things a bit higher, or a bit bigger, I should say, not higher.
But even if I don't understand it, I think it's fascinating when y'all go over stuff like that. Dang, remember the hangouts y'all did on what's his name? It was claiming there was a functional gene across the fusion point.
Tompkins,
yeah.
Well, that's been an interesting thing because in Ronnie's hangout, or in G-Man's hangout earlier today, Ronnie was in the live chat complaining that apparently we don't respect Tompkins' credentials enough, which is interesting because Ronnie never respects our credentials. So either the arguments stand on their merits or they don't, Tompkins doesn't know what the fuck he's talking about when it comes to this stuff, or he does and he's being dishonest. I mean, those are the two options.
Because one thing that's so cool about all those hangouts is like y'all took his information. It's okay. Well, let's check these databases and stuff. And you're trying to see if y'all can find what he found, or if these databases show what he found. And then y'all got the site for the actual fusion, and then from there you can actually go and see in the database. Here's the fusion point. Here's all the stuff that's on both sides of it.
Well, he didn't have a method section in his
write-up.
Right. So like we had to recreate everything.
Yeah, he didn't have one. That's why they spent 4 or 5 hangouts on all those databases.
Well, because he used different coordinates from different genome builds. So I had to go and I had to convert all of the coordinates into a different genome build. It was such a pain in the ass. And like Hogtie was like, I'm not finding any of this stuff. And then I was like, oh no, it's because he's making this idiotically hard to understand. Like, no shit, you're not finding anything here. It's because he's using a different build of the genome than we are. It's such a pain in the ass.
I think that was my one and only contribution to all y'all's efforts, was remembering that, oh gosh, what's his name? The Catholic guy, Ken Miller. That in one of his videos, he actually gave the site for the place for the fusion. Okay, I got this.
Yeah, I didn't know. You said he didn't have Tompkins didn't have a method section?
No, sir.
What? How? Wait, hold on. Okay, so if he wanted anybody to believe him, anyone who's not a young Earth creationist or an idiot, why would he think that foregoing a method section was a good idea? That to me is really baffling. Like what? And this, I guess, this speaks to, I guess, True's ability to understand what the problem is and why I guess myself and any other person who's ever read a fucking paper has a problem. Like, if you can't show what you fucking did, why are people supposed to believe you did anything to begin with? And how? Like, that's frustrating. Like, that's really frustrating. So folks who are listening, the fact that this dude has just completely foregone this, making a fucking method section, I think just on the face of that alone kind of shows you the caliber of a person you're dealing with here. Because what are you supposed to do with that? Nothing. You can't do anything.
I don't know, right? And he adds in that there's like a site of transcription initiation there, but he never talks about the fact that like we don't know how to interpret that. It's a whole mess of nonsense, to be honest, because he doesn't give principled reasons for why he thinks these things are functional. He just goes, "Oh, look, there's this stuff here." Well, great. It might be functional. I don't know, even if it is functional, how would that actually interfere, since there's been millions of years for these two species to diverge? But he doesn't care. Like, I feel very comfortable saying he's just a dishonest person.
Well, and also, remember Rubio got in touch with him and corrected him on something? And I think he actually retracted part of
No, he doubled down.
Oh, he doubled down? Oh, okay.
He retracted something regarding the it wasn't regarding human chromosome 2 fusion. It was regarding when he was comparing humans to chimpanzees genetically, because there was an error in the BLAST tool. And it's one of those things where why would you you shouldn't use BLAST to compare two genomes, because BLAST is a local alignment tool. And you're doing a global alignment, but I'm also expecting a creationist to be logical about these things. And the reality is he doesn't know shit about bioinformatics, I'm guessing. And he doesn't know how to do any of this shit.
He didn't have any bioinformatics students working for him?
Do you think he has students working for him right now?
Good point.
Like, how would you like to work for that to increase your resume? You're a student.
You're funny. No. Oh no. Oh God. Jeez. No. Molecular interactions. I'm trying to, like, Nethar wants me to look up a course. It says molecular interactions.
Check what I just wrote in the Hangout invite chat.
Oh. Let me pull it up.
You can tell me if that's appropriate or not.
Loading.
Fitting?
Oh yeah. Oh man. That's about 1,000 years of hate right there, just for that one comment. That is 1,000 years of hate, sir. Oh God. So
it's a fair question.
It is. It is, and
it's a fair analogy.
It's a special kind of love, to say the least. So I'm looking right now, Nethar, at all the chemistry courses offered at my college. It's going to be kind of limited because this is like technically like a satellite campus. It's not the main campus, and I'm not seeing, like, I do see. Where is, I just saw Life in the Universe, Chem 2377. I think I wrote a little bit about this, or I mentioned it a while back. It says no soil and groundwater remediation. Let me see, life in the universe. Yeah, I think this is a course I mentioned, the hog tie.
Who is Super in your chat?
Oh, he's one of the kids that was in the off-air chat where I engaged Nephilim Free that prompted him to make that video.
Yeah, Super does not know what he's talking about.
Well, I think
he
that's
said that the lipid bilayer is comprised of proteins. No, there are proteins associated with the lipid bilayer.
Yeah, you have transmembrane proteins. I think this is the kid. I thought it was like Super. I think I had a number in it too. No, this might be somebody else. I don't know who that is.
Well, Super, if I gotta say, if you're gonna complain about your use of language, note that capitalization is the only difference between helping your Uncle Jack off a horse and helping your Uncle Jack off a horse. So it is actually quite important.
Okay. Oh, so Nethar, I don't know if you're still watching, man. I don't find that class that you had mentioned about the chemical interactions. The closest thing is Chem 2377, Life in the Universe, and the description is an account of the origin of the universe, origin and evolution of life, and the possibilities for finding life on other planets, including the search for extraterrestrial intelligence. That's really interesting.
No, phospholipid is not a fat, Super. You just stop. Oh, God.
Since not many university classes have classes like that yet, but they should. Biophysics is really important nowadays. Well, there's still a lot of campuses that are trying to incorporate. I think what you're talking about, Nether, are like interdisciplinary courses, because I have seen a paper on the kinetics of intracellular processes, I think it was called, and they do focus on the kinetics and or whatever within the cell, so within and I think outside the cell. So yeah, there are more people that are kind of jumping on that, but it's a slow progress, because how many people are going to be willing to learn both physics and biology, just like fucking Sam's field, computer science and biology. Not a whole bunch of people jumping on that bandwagon, but they should. Phosphate head, glycerol, fatty tail. Okay, he's describing the fucking.
It's a fatty acid tail. Like, you don't say that it's not made out of fat. Fat is comprised of lipids and fatty acids. It's not.
Like, is he confusing adipose with fatty acids?
I think so. I just think he doesn't know what he's talking about.
Yeah, there's definitely a difference between what people colloquially call fats and fatty acids. There's a big difference, because you do have lipids involved in what we typically call fat, but that they're not one and the same. You have to be very careful about what you're saying, and then that goes to the point of when True and some of these other guys are trying to speak science talk, they're trying to use science jargon. It is very important. That's why I can't call fucking cysteine cytosine and just okay, yes, whatever. No, they're completely different things. Yeah, you're Neto. If you don't mind, where is your university? Because you have a course like that, man, it must have a little bit of money. Not no rinky-dink fucking university got courses like that. Well, then again, there's
well.
Also,
I compared Blue to Hitler, and Kevin said Blue's an ex-crackhead with anger issues. I don't exactly see them on the same level. And I'm going to come back with competency aside, both murderous bastards.
You guys are going to get so much hate.
I'm gonna get so much hate for saying that. But you have to say it's pretty funny. And
it was funny.
I don't think it's that. I think it's pretty appropriate.
Oh my goodness. Dragon, I got lucky. My university had a lot of cross-discipline classes. We had classes on physics and programming. Oh, that's interesting. As well as usual biology and chemistry. Yeah. I don't think that has anything to do with luck, man. I think that has to do with money. Because if you have instructors that can teach shit like that, man, they've been attracted there for a reason. I don't think that's luck. You got a robust program going on. Also, our math prof was from physics engineering faculty, so he used a lot of physics examples in his math got me saying meth. God damn it, math lectures. And see, that's what I like. I like people who are able to incorporate that, within the realm of teaching. So it's not just people coming up with, well, Susie has 6 candies and Timmy has 12. How many hours until Susie gets murdered for a candy? Like something that's more relevant. Westphalia, University of Applied Sciences, Westphalia. I think German education system. It only costs what is that? €226 per semester. Well, fuck you.
Fuck you.
I had to spend 4 years of my life fighting for this country to get my education. Fuck you, sir. I'm actually on the school website now. Welcome to Westphalian University of Applied Sciences. We are a medium-sized university of applied sciences in one of the most active regions of Germany. It's a front page normally in English, or did it translate? No, I don't think it is translated. Wow. It's in, medium-sized campus. Looks like a good fucking school. Props to Nathar for that.
Yeah, my undergrad cost a fucking arm and a leg.
Still trying to grow them back now.
Well, I mean, I had some smart investments and got some partial scholarships. Like, I got a full academic scholarship for my master's. So that helps.
Yeah. Oh, yeah. Oh, yeah. Yeah, I don't know who Super is. It's not the same person I was thinking about.
Yeah, he's just a jackass. He's like, you're being arrogant. I don't fucking know what you're talking about. I don't even know why he's going on about membrane-associated proteins and membranes. I don't know why it's relevant here.
Well, the second he starts talking about, G-couple proteins, then I'm just going to turn the headphones off.
Maybe it's just Ron. It could just be Ronnie.
No, he actually knows how to use punctuation. It's not Ronnie.
Ronnie's been getting better lately at that, though.
Has he?
He has. He hasn't been making as many grammatical errors to the point where I can't tell if he's trolling sometimes. Let's see who Super is subscribed to. Go to channel. It doesn't look like it's Ronnie.
Nope. Nope. RSC says the arrogance
is just a normal tone. Yeah, it's just normal tone. What are you? You said something stupid. I pointed out it was stupid. What's the problem here?
Oh, God. Oh, this is the shit.
I don't understand people. They're like, I want my ideas under scrutiny, and then you're too harsh to them. I don't want my ideas under scrutiny. Make up your fucking mind.
Can't have it both ways, right? You guys should get with Fiona and Jay and do a hangout. What's considered, what is considered life and what isn't, especially viruses, prions, point out how a living God is absolute nonsense. That would be a pretty interesting hangout. All we would need is a virologist, and I mean, we'd have a fucking fist fight practically. Because like I think virologists are the only individuals who actively promote the idea that viruses are life. Everyone else are like, no, they're not.
I love you, RSC. RSC just goes, JK Sam, I love your direct approach. Thank you, man. Well, Drag knows me off air. I'm this blunt normally, so it's not.
Nothing changes. That's just him. That's why I'm baffled why Gmail was like, Drag, now you changed. I'm like, no, the fuck I have not, dude. What the fuck?
You're the same off air.
Like, I don't. I'm trying to figure out what he was talking about, because when I got there, he was like, "Wow, you really are the same." I'm like, yeah, I've told you that, and so now, oh no, you changed. Like, well, make up your fucking mind. Am I different? How am I different? Was I? He says recently. Well, what is? Okay, you kind of see. I'm a little different. Okay, so how? Okay, so they'll tell me, dear, how am I changed? So I'm listening.
Did he get the superpowers granted by being my hashtag token black friend?
Wow. Felt like you don't have the passion you used to have making your videos. Like it's just kind of like
I don't have the passion.
I'm just doing this for shits and giggles.
Oh, okay.
I'm really doing this because I want to do this. Or I have any interest in it. So I get what G-Man.
I don't have the passion anymore. I don't have the vigor.
Right. Like,
I'm too busy using it at night.
Oh.
No, I'm joking. Okay, that's fine. Okay, I understand. I think you're giving G-Man a little bit too much credit, but that's fine. I'll take that. Be passionate. That's what people wanted to hear. That's like the only reason I even kept doing it is because people wanted to hear about it, and that's it. It's like I got tired of repeating the same story over and over. But I'd rather talk about science. Yeah. Oh yeah, he was mad at that. Yeah. Well, that's fine. I mean, he can feel some type of way about me taking his audience. Well, who's more entertaining, me or G-Man? So it's a competition.
Well, G-Man has his moments of being very, very entertaining.
I agree. Well, yeah, I told him he can roast me. Yeah. He's like, I get a chance to beat him. I can beat the final boss finally. And so, yeah. I know his weakness. I shouldn't have allowed him to do that because he made me burn my roux yesterday, and I wasted my roux. You say, I know you get all the empathy. I understand.
Oh man,
what's
I just saw a photo. People are such garbage. There's someone who posted on a picture of a trans person wearing a shirt that says transgender veteran. I fought for your right to hate me, and the first comment is nice tits, bro. People are such garbage. That
would be the same guy, the same exact guy that would be in that person's inbox. Because I'm joined this. My wife and I both joined this crazy fucking group. I'll tell you what the group is in the side chat. But there's a bunch of trans women in there, and they're all from like the Houston area. Some of them have fooled the shit out of me and my wife. No, neither one of us could tell that it was a trans woman, right? It's a beautiful woman. And so there'd be guys in there. Oh, that's a dude. Look at all forearm strong as hell. Look at that. And so pointing them out. So okay, whatever. And then the person like, yeah, I'm trans. Okay, so what? But then what happens is that after everyone has their turn, saying whatever disrespectful remarks, a lot of the supposedly straight dudes would then go and add that person. And so what that trans woman would do is like take a picture of it. And all those friend requests from all those folks, and then all the comments, and put them side by side, and then post those in the group. So those same fucking guys who are talking so much shit secretly wanted to get them. I thought that was pretty funny. It's a very ratchet group, to say the least.
That does sound pretty.
It's very ratchet, very, very, but it's funny as hell. Can drag down. I have a video of me dancing to one of G-Man's videos like a few weeks back, just before the storm. I was just doing stuff. I was working out actually, but I was doing interpretive dance.
So in that chat, so Kevin typed, if making a death threat on the internet was equivalent to actual murder, I don't know if we'd have enough space to imprison everyone. So then I wrote, can we enact that for Blue? I think the butt sex in prison would calm them down. And then I typed, Nicola, you're the resident expert on butt sex. What say you? And he says, in my experience, people don't make death threats if they're biting a pillow. So...
Oh man,
I'm not a good person.
He has a point. He has a point.
Yeah.
So Dragger.
No, I don't think anyone wants to see you do a dance off with G-Man. That's just a lot of.
It's a lot of moving parts.
That's a lot. It's a lot.
That's okay. I'll pass. Was it Dragnout? So Trap's gay. I don't know. Do you? That's my policy.
What's a
trap?
What's a trap? Like a trap? Like a? Is that a trans person that looks like the opposite sex really convincingly, or?
Yeah, basically. Like if they entice you, but it's like, oh, surprise, penis. They call it.
That's not that's not cool.
I
would say that if you surprised me with a penis, I would not be pleased. That's not like a lighthearted birthday surprise right there.
It's like, oh guys, you got me. No, there's a dick in there. Oh. I don't know. I've seen that question posed in many like hentai and anime threads that aren't hentai and anime thread related. So it just comes up for some reason. Like, I don't know. I don't care. Like, if you like trans men or trans women, fucking go for it. It doesn't matter what anybody thinks. There are some trans women who are fucking beautiful. I don't care what anybody says.
There are a lot of trans women that are better looking than Shani. I'll tell you what.
Oh God. Oh fuck yeah. Jeez.
Most of them.
Like they take pride in how they look. There's a big difference.
Well, Shani does take pride in how she looks. That might be the problem. Is that too mean? Is that too mean?
That's just right. Happy Jesus says Tinder is 32% trap, 33% fat.
I don't agree with those numbers, but I'm also in Southern California, so
demographics are a bit different.
Yeah. I don't know. You've seen some of my recent Tinder matches.
If some of those are traps, you know what? I'll be a trap motherfucker. I'm just saying. It's like you got me every time.
Well, remember her name was Taylor? I never ended up meeting up with her. That was a bummer. We were going to get wine. It's my specialty.
Oh, the really cute one? Really cute one?
She wasn't the really, really cute one. That was just trolling for Instagram followers.
Oh.
The one where, like, if I hooked up with her, I've accomplished everything in life.
Yeah. You could just...
Cannot retire yet.
Hey, the hunt is on. Expert in the topic, 1,000+ hours of field work. You know what, man? If you want to dig through the weeds, so to speak, hey, to each his own.
Hey, sometimes you gotta take a few strokes in the rough to make it to the fairway. You get what I'm saying? Oh, it's a rough one out there. You know what I'm saying? I don't think I've ever hooked up with a trap.
You might have and not just know. It's possible.
I feel like I would know.
I gotta find the pictures because I sent some pictures to someone last night.
Well, no, I meant like once the pants come off. I feel like I would definitely know.
Oh yeah. Once the jig's up, yeah.
I'd be like, you've got like a really protruding wallet. I don't have a wallet. I have to leave. Like,
can still be friends. I need a wingman on the weekends. Actually, you know what? A trans woman would actually come in handy for that.
No, I have plenty of like cis women that can do that role. Although my best wing woman she is living in Singapore, so that is unfortunate.
It's a little bit of a. It's a little bit.
She was like, it made it so easy because she's gorgeous, and she's Scottish. So people are like, why are you walking arm in arm with this gorgeous Scottish girl?
Reasons. Just reasons.
Just reasons.
Okay.
So I'm about to send you. She's the blonde in this picture.
Bloop. Where is it? Oh, hello. Like a news reporter.
Yeah, she does look like a news reporter.
Nice.
Shani is trying to trap G-Man.
Can
I get that G dick in her with that G batter, and then make a G child?
Lucky she didn't make a video about you this morning.
I would rather rip my penis out by the root than see Shani naked.
That's just me.
It's true, though. I think Super says he wants a link.
Sure. I'll put this copy.
Oh, God. Happy Jesus. Too much. It says, Shani is going to duct tape G-Man to the bed and have her way with him. The hilarious thing is G-Man doesn't recognize the signs or believe the warnings.
Well, fellas, there's not much more I can add, so I'm harassed, so I'm going to head on out.
All right, Heavy. Always good talking.
But thank y'all for presenting that, though. I appreciate it.
Oh, not a problem. Not a problem.
Hopefully we'll pick a paper that is a little bit more accessible to both of us. I don't know the nice words of maybe we should know what the fuck we're talking about.
Hey, Sam.
Yeah.
Oh, it is him. Okay, yeah.
Yeah, this is Super. I just mean to say that I guess I wasn't very clear about my language and my terminology on the chat. And also, I guess maybe in the process I've been corrected. So, I mean, I guess for like to be intellectually honest, I guess I'll just go on and say that, hey, maybe that that was worded, I guess, you know. But anyways, yeah, I just meant to say that the phospholipid layer, bilayer, it's not like comprised of proteins, but that in the system of a plasma membrane, there's a protein, like there are proteins and there's many things inside the plasma membrane. You know, that's all I just meant to say.
Okay, does that connect to some larger context?
Perhaps like I guess I just wanted to maybe use chat as some sort of like so people could correct me, or like some sort of learning strategy as a way, like, you know.
Are you just now learning this and you were trying to talk about it, or?
Yeah.
Okay.
And I don't have a book with me, so it's like I'm doing this on top of my head. So. I mean, I did learn about this, but it's just trying to put it all together.
Well, stuff in this paper is because it sounds like
beyond that and a bit unrelated.
Yeah, that the stuff like you're mentioning sounds like intro bio stuff. The stuff that we're talking about is it is on it. Like you can find some of it but it's like in a principles of biology class, the chemistry of life section or whatever they want to call it. But yeah, it's not really related to what's happening. So congratulate you on learning this stuff though.
Yeah.
I mean, it's kind of like review, but I don't know. But I just don't mean any animosity, I guess. You know.
No. There's no animosity. Just it's being. I think that's placed emphasis on being clear, being very clear with one's words, because when you are talking about stuff in science, you can't be very sloppy with the language, which we've talked about before with certain people in our community that just kind of just say things and they don't really understand what they mean, but then they get kind of belligerent when folks go, "What?" Yeah.
Yeah, true. All right.
But yeah, I don't think anybody
Anything else they want us to address in the live chat?
Give them a
Before we
few
take
minutes.
this?
Yeah, we'll just take it off air. Anything else, folks? I think because I'm starting up my intermittent fasting stuff again. I have my coffee in the morning, water throughout the day, and then I'll probably wait till 6:30, 7. What's up?
I'm not sure if you know this right off the bat, but I guess I have a question, and it has to do with you know how there's active and passive transport on the membrane. So I was wondering, there is active transport, and then with that comes what is it called?
Exocytosis?
Yeah, exocytosis and endocytosis. So I was wondering, you know, how facilitated diffusion, which has to do with passive transport, what exactly is the difference between facilitated diffusion and endocytosis and exocytosis? Because I know I think that they all use protein receptors, if I'm correct.
Well, passive transport just means you don't have to put energy into the system to move the components. Whereas active transport, you need to use energy. That's the main distinction. There are varieties of kinds of endocytosis, but I don't.
Yeah.
Is there a greater context to that?
Or are you just kind of going over some of the basic stuff that you had just learned?
Yeah, pretty much that.
Okay, so if here's a trick. If you're not sure what anything means and you're having trouble discerning between one thing or the other, break apart the words. If you write down or look up endocytosis, you can break it up endocytosis. So anything, I would say almost anything with the beginning of endo, something's going inside of something. Exo, something's being excreted or going outside of something. Anytime you're talking about cyto something, that's something to do with a cell. Well, I can't really. I need to be more specific with that. That's somewhat right, but not the most accurate.
Yeah.
Let me look at the Latin root.
Well, I mean, endocytosis is just the taking in. It's an active transport where you take a chemical in. Exocytosis is when you excrete a chemical using active transport, whereas passive transport, you don't need to put energy in, so it's usually through some open pore, or it can diffuse across the membrane barrier.
So I'm trying to think of something that will require facilitation, like what is it called? Sodium ion channels, I think, have to be facilitated.
I think so.
Yeah, I want to say that they can't because
You mean the
Or do you mean the sodium potassium transporter?
Yeah, the sodium potassium. I think that has to do with active transport, based on.
Because the sodium ion channel, that's passive transport, I'm pretty sure.
Just passive? Oh yeah, because of its charge, it's able to diffuse. If I need to look that up again. Yeah, because I think you're right about that.
Floyd has presented a knockdown argument for creationism. If evolution is true, explain how nature selected for G-Man.
Well, folks, it was good. Good night. See you later. That's actually pretty funny. That's wow. So I'll teach you a trick, Super. If you have trouble remembering this stuff, man, learn the Latin roots to spell out what something is and have examples. That's why I went to the sodium ion channels, because the sodium ion channels and the sodium potassium channels differ because of how they get across the membrane. So if you can, relegate them into active or passive, that'll help you then remember which one is which. Does that make sense?
And also, is the word ion or like its function, the ionic pump, the ion pump, I believe, is that word ion like indicative of active or passive transport?
I mean, it's just a charged particle. Like I'm not sure. This is getting a little bit more into orchem than I care to speak on. But Sam, are you able to answer that a little bit better?
Well, it has nothing to do with what type of transport it is. It has to do with the molecule being transported.
Okay.
Like we just discussed that a sodium ion channel is passive transport, but a sodium potassium transporter is active transport. So it just depends.
Yeah.
How old are you, Super?
I'm not 12.
Okay,
No,
are you 13?
No, I'm not 8 either.
Okay, motherfucker, what age are you? I don't need to know. You are an infinite. I also know you're not 77 years old. So what?
I might be that age. No, but okay. I'm 21 though. And I'm just reviewing this stuff, I guess. And I guess I just meant to write it down in the chat and stuff, and I guess I had it looked at and stuff like that. So.
If you want some practice, man, yeah, you go to work on your social skills. Go to Neffy's channel and start with the shit that he says, and then start learning about the topics that he's trying to mention and learn what they really are, so that one day you can talk to Neffy and realize all the stupid shit that he says. Does that make sense?
Depends on which topic, because I can really break down his topic depending on which specific one you're talking about.
Well, pick one that's close to, like cell biology, especially things that deal with, abiogenesis.
Hmm.
I think topics close to that will be somewhat related. Close enough for you.
Yeah.
How you doing, Pseudo?
Headless chicken.
I don't think his mic's working again. Pseudo, mic check.
RSC thinks you just gave terrible advice.
Well, RSC can eat a dick.
You heard it here first. Start going nom nom on that penis.
Look, if he can go to Nephilim Free's channel and listen to it and try and figure out what he's referring to and then go learn this shit, he is preparing himself to read on the origin of species. I'm not lying.
Oh, apparently James Damore was on the Joe Rogan Experience.
Is that the fucking conspiracy nut?
No, that's the guy who got fired for writing the sexist memo.
Oh, okay, that guy. That sound familiar. Oh. Heard he got a bunch of job interviews and shit. So
good for him.
That's
I mean, if they want to hire someone who, believes that stuff, and more power to them.
Free market and all that shit. Midget strippers. Oh my god. Anyway, Neff kills brain cells. He does. No, he does.
He uses a lot of whiskey and Coors Light.
He kills his own brain cells, so you don't have to. Oh man. Okay, I guess Neffy or not Neffy. I guess Pseudo's reloading. Oh, if you can sit down and try and listen to the shit that Neffy says, it's commendable.
Is that the guy that came in like weeks back?
Yeah.
I think I was there.
Yeah, that's where I think that's where you after you subscribed him. Whoa, what the fuck? G-Man went live. Let's talk about dogs and evolution.
Oh boy.
Oh, he disabled his live chat for this. All right, let me play a little bit of it.
Is he live right now on GTV or on?
He was on GTV and he was alive like 18 minutes ago. There's no description.
No, he already went back down. Why didn't he join us?
He knows better. He knows better.
Is it because we have no white women here?
Secret's out, folks.
It's our lack of white women. Shit.
We gotta have Helena just on hand. It's like, look, I just need you to sit there. You don't have to say anything. Just have to be there.
Well, the question would be is, who's his type, really,
Are they white?
Evidently.
No, they're women.
I don't think Shani's his type. Because I'm gonna level with you, Drag. If she was hot as fuck, you would have been like, oh, well, praise Jesus, this is fate,
Oh yeah. He would have been to where does he live? Minnesota. Yeah, you'd have been there in a heartbeat. Yeah.
So definitely not her.
Get my message about G-Man. Jared, where did you send me a message? Did you send it on? I didn't get any messages. Did you comment on a video? I'm looking at my comments now. No, I don't see you. Like a belief in white women. Okay, my wife. What is he saying, Tasha? What is he saying? Okay, I'll play a little bit of it. I'm gonna play all of this. It's 17 minutes, but I'm gonna play all of it. I'm just curious.
Hello, everyone, and welcome to GTV. I'm G-Man, your host, and today.
Oh, mic check, Pseudo.
Greg is the king of the black people.
There we go. It works perfectly.
I want to talk about dogs and evolution. I've been in a few hangouts here on YouTube and spoke to quite a few evolutionists on here who claim that if a dog becomes a non-dog, that would not be considered evolution.
He finally gets it, folks.
I do want to say for the sake of this video, I will not be getting technical in this video. I will make this so simple that even a caveman will understand what I'm going to say here today.
What's that old
Obviously,
Geico commercial?
I do not agree with that statement. And I'm going to show you why very
I
soon.
think that was a Geico commercial.
All right. So, again, I've been in some conversations with some evolutionists. I've listened to their theory, looked at their theory. I'm preparing a debate right now to debate someone on the theory. And every now and then I'll run into a person in the room that laughs at me when I say, well, if you believe in evolution and you believe a dog will eventually evolve into a non-dog. And what they try to do is they try to make me sound like I'm saying that a dog is just going to give birth to something that's no longer a dog.
That's what you're saying.
Right? That's not my claim, that's their claim. You see, in order for evolution to be evolution, there has to be mutations, there has to be changes to the environment, to a bunch of different things. And if a dog is having sex with another dog, half of the DNA of the mother dog, half of the DNA of the father dog is going to produce another dog, but this dog is going to look a little different, it's going to act a little different. It's going to take on some of the characteristics of his mother and of his father. I have a cat here that lives in my house. If that cat has sex with another cat, some of the cats are going to be black and white. Some of the cats are going to be the same color as their mother. And these cats are going to act like a little bit like their mother, a little bit like
that better be a cat if he's holding something up.
Had kids.
He's not holding anything.
It's the only pussy he's going to have in his hand anytime
I'm saying it still better be a cat. It better not be those things he found in the
Oh,
park.
gonks.
And half of the characteristics are going to be instilled in the son. I'm sorry, it's going to be instilled into the child. We got no argument there. That's called variations within the kinds, and I don't have a problem with that.
Kind.
What
What
is a kind?
Term evolution, where some will argue that the first living.
Actually, let me pause it right here and screen share G-Man's actual definition of a kind. Hold on. Y'all remember what it was?
An animal that.
That is the same kind of animal, isn't it? Something to that. It's some really circular definition.
Where is it at? It's somewhere.
All right, I need to take off. Thanks for having me.
All right, I'm just gonna leave this open a little longer, then I'm gonna take off myself. Okay, this is G-Man's definition of a kind: an animal that's like the same thing as an animal.
What?
And the source for this, folks, is here.
I don't understand. Now I want to get to the definition of what a kind is. A kind, the way I define it anyway, is an animal that's the same thing as an animal. Like, for example.
Okay, just so we're clear, folks. That's his. I went to the link, read the quote, clicked the video. You heard him say it. That's his definition of a kind. That is the most nondescript description I've ever heard of an object or a thing or concept. Excuse me. I don't even know what that means. So there we go. Just so everyone's tracking with that. All right.
Was a tetrapod, and this tetrapod, small changes over a long period of time. A tetrapod with another tetrapod, because they was able to mate with each other, had a tetrapod with small changes, and it looks slightly different. And if you do this for millions of years, according to evolution, even though I never seen this, I gotta take the word for it. These changes over a long period of time, you're gonna have something that no longer looks like a tetrapod. So when you hear me say a dog is going to become a non-dog, that's what I mean. And believe it or not, the person that I'm talking to knows that. You're playing a game of semantics at that point, where if you don't say things exactly the way the book is saying it, that somehow you don't know what you're talking about.
Well, it's not an issue of him not saying it is like verbatim. It's him completely messing up the concepts. Like, there is no, G-Man, you didn't quote this particular book that I have in my hand. Like, no one's ever made that stipulation to him ever. It is that he doesn't understand the concepts, and then he uses shit like kind, which there exists in no study and in no book, and no one actually uses that because it's not descriptive at all. So he wants to be able to straw man shit, and he's upset that we point out that he can't straw man. So now his new turn of phrase is, well, that's just semantics, without actually knowing what that means. So yeah.
Has anybody ever tried to explain evolution to G-Man like a family tree?
People have. People have tried to use that and point out that cousins are still related to you, but he still doesn't get that because you still look like your cousin. So just for reference, folks, tetrapod, we're still tetrapods, okay? I said tetrapod is super class, all right?
Wasn't G-Man the one who got upset about being called a biped?
Yep, I called him a biped and he got upset. Tetra 4 pod feet. Okay, four limbed in this case. Look at that. See, amniotes. Look at that. Class mammals. Oh, we're a mammal, aren't we? Yeah, look at that. So if
look at that! Wow.
if I click on amniotes, man, look at that shit. Man, it involves comprises of reptiles, birds, and mammals. Let's see what's in mammals. Oh shit, man, look at that. Oh, people are in here. Look at that human.
Oh, look at that.
Fuck me. Look at that. Okay, so yeah, so we're still tetrapods, folks.
I won't have that problem in this debate. I assure you of that, because any technical statements that need to be made, I've already got that covered. I'm actually memorizing terms. I'm going to say it as it is in the science book.
Then stop saying kind.
Say that that's not what it say. I can literally show you like what it says. So does a dog becoming a non-dog prove that evolution is not evolution? I say,
Greg, somewhere in North Korea, G-Man videos are being used as a form of torture. You know that?
Yeah, but you're getting it for free, so you should feel lucky.
Yeah.
Yeah. What's the use of saying kind anyway? Like what's the whole purpose?
It's just bad language.
So, since you just kind of learned this stuff, let me go over it. The reason they want to use kind is because that's the same language used in the Bible. That's the only reason why they insist on using that nondescript, because they can't. If he were to say I'm using kind in place of species, I wouldn't have a problem with that, because I know what a species is, and there's a certain delimiting factor as to whether something becomes a species or not. We have a criterion. Yeah, if you, I don't know how far you've progressed into your education, but you'll learn about biological species concept as opposed to the phylogenetic species concept. There's a different criterion for each of them. One's better than the other. So that gives you certain stipulations on these labels and all these categories. Kind doesn't have a category. It's just what the word kind and the categories within that are only for colloquial uses. If I ask you what kind is a lion, oh, it's a cat kind because it looks like my cat. What kind is a platypus?
It's a duck beaver.
Can I toss two things out real quick?
Go for it.
One, tetrapods also include things like snakes, G-Man. Even though snakes do not have 4 limbs, they're still categorized as tetrapods because they evolved from tetrapods. As again, you don't lose your historical taxonomic levels. And when you keep saying a dog can produce a non-dog, that's the same reason why we are still mammals. All our descendants will be mammals. All the descendants of any tetrapod will be tetrapods. All the descendants of snakes will be snakes. This is not a complicated thing. And also in the Bible, actually it does not use the word kind. The original Greek Septuagint, or one of the earlier manuscripts, actually uses yevos, which means genus. It just meant family or descendant. Kind wasn't actually added in until later. But of course, being King James only literalists, or people that think that the later version somehow superseded the prior versions, they have this kind that they have to work with now. But the original manuscripts, the earlier ones that are available, it just meant descendant. That's all it meant. So now they have this weird word kind, which is ambiguous, has no real meaning. And if you ask 2 different creationists, you're going to get 2 different answers of what a kind actually means. It's going to be anywhere from species to genus to phylum to kingdom to domain. Who the hell knows what it means?
And that's the biggest issue with this, is that you can't be ambiguous when you're talking about classifying things. That's literally the antithesis of being being specific. Like you want to be as specific as possible when you're classifying anything. It can be marbles, it can be rocks, so offering something that's vague like this does not work for this purpose. So let me play a little bit more of this, because I don't know if you're going to start a hangout, Steve, but I'm going to end this.
No, I'm working on another math presentation. We're going to be doing composite functions because we all love those, right?
Oh, are you trying to segue into chain rule?
That's exactly why I'm doing the composite functions first, because I figured, how are they going to understand a chain rule if they don't know what a composite function is? So
as soon as you said it, I'm like, you're getting in a chain rule.
Are you trying to do that today? Exactly what I'm doing. You got it.
You doing that today, or are you going to?
No, I don't think I'll be able to finish it tonight. Maybe if I finish it, but I'm not planning on it though.
Okay.
It takes a lot of work to type this into this math program.
Yeah. I guess I just wanted to chime in, like how it seems I find that people trying to modernize the Bible has maybe a sort of opposite effect, as does scientists or people who research in evolution, try to find new things about evolution and kind of produce new and different results
Huh?
that they've done before. So it's
No, super.
they're sort of,
What you're kind of missing here is the Bible is not a science textbook. It never has been. It never will be.
I don't think that's the point that I was making.
Okay, it kind of sounds like you're going there because
Uh,
you're making no
The
sense.
point that I was making was that the Bible, as we know, like there are new versions of it. So what I said was that that the Bible is modernized, and that
Oh, you mean the translations are cleaned up?
based on like messing up the whole interpretation, or are kind of compromised, and as such, the interpretation of the text becomes what people like. The new versions are what people seem to base their understanding on the Bible. And I was saying that this whole modernization of the Bible is sort of opposite in terms of um, when compared to scientists finding new things about um, a certain animal per se, or like species. You know how
I
science.
have no idea what you just said.
Yeah. You were using words. I got that much.
Yeah, I think what you need to do, maybe Super, is kind of like parse what you're going to say out and try to put it in a way that makes a little bit more sense, because you're not really taking us to where you want to go. What you think your argument, or actually what you're saying, makes any sense, because it really doesn't. So I mean, for start from the beginning here, do you accept that kind has no meaning in taxonomy?
No.
Okay, so you think kind has a taxonomic meaning?
No.
Okay, you just said no to two different questions.
Oh wait, what was the second question? My bad.
You do or you do not think kind has any meaning in taxonomy?
No.
Okay.
Based on it being vague.
Okay.
What was the second?
You're not a Bible inerrantist. You're not a young Earth creationist, right?
I don't think so.
Okay. So you're not a big fan of Nephilim free, or?
Don't think so.
Okay. You don't think so. You're not sure. You may be a younger creationist. You're just not aware of that if you are or not.
No, I'm not. I don't see any basis for it.
Okay. Are you an old Earth creationist?
No.
Theistic evolutionist? Atheist?
Yes.
Okay. Okay. Boy. like pulling teeth here. All right.
I can tell you everything he's not, just not what he is.
Yeah, exactly. I know. Exactly. There's an infinite amount of possibilities. I'll eventually hit the right one, right?
That's odd. All right, so let me play a little bit more of this.
Has sex with another dog, and they produce a dog that has characteristics of his mom and characteristics of his father. Some have bigger ears, some have smaller ears. And let's say that dog has sex with another dog and took on characteristics and had another dog that looks slightly different from his grandparents. And let's say those dogs had sex with another dog. Let's say they interbreeded with another kind of dog or whatever. Them dogs are going to look slightly different, right? According to the theory of evolution, right? Now, again, I don't have a problem with variations with the kinds, because I know at the end of the day, you're always going to produce a dog, a canine.
What we're telling you,
right?
G,
It's
man.
slightly different from the great grandparents, but you're always going to have that dog, that canine.
Well, he's getting
in order
it
for the
right.
theory of evolution to be true, at some point, that canine has to become something other than a canine.
Why?
I'm going to show you a little picture here. I'm going to show you guys here.
Why does he keep thinking
I went
that?
to the Museum of Natural History, by the way, for those of you who will watch this later. So I don't think nobody's watching right now. And I had an opportunity.
Yeah, nobody watches you. Man, that's
Well, he said he didn't want anybody to watch that channel. I'm not sure why he's surprised.
And even the skulls is telling the story the way I'm telling it right now. Now, I'm not using a lot of the technical mumbo jumbo that a lot of scientists will use. I'm not using a lot of that jargon.
Or the technical mumbo jumbo that Ronnie
If a
uses.
child watches it, they can understand what I'm saying. And that's important. Not important for me to boast and brag about how much I know. It's important that you, the audience, understands what I'm saying.
I think I agree. So I completely agree.
That's important to me. So anyway, I'm going to screen share something with you guys here.
Can't wait.
With, I think I'll just show you. Hold on.
I just want a coherent argument.
Would be kind of nice, right?
That's it. Like, I don't want anything else.
With anthropology.
Oh, look at the family portrait. I see G-Man.
The 8 family tree, human evolution, genetics, I think. The 8 family tree, human evolution and genetic
Pinterest.
Anthropology. Okay. And what we have here, even though this might be a bit goofy, we'll put this together.
See, it's a cladogram.
I can find something a little bit more serious.
That's a cladogram.
This here, just make you more happy. That's goofy too.
Goofy cladograms.
For the sake of the conversation.
Damn these trees.
Guarantee you he doesn't know how to read this.
Oh, guarantee he doesn't know how to read cladogram. Because it's not the same as a phylogenetic tree. It's about traits. You need character combinations.
The reason why we know that a Homo sapien is not a monkey, a monkey is a primate, and a Homo sapien is a human. We know that monkeys and humans are two different.
Pause it for a moment, Drag.
Sure.
Two different things. I'm gonna put it that way.
So on this family portrait, where's G-Man?
He's
third from the right.
Third from the right.
Oh God, I don't even want to know where this is going to go. This is scaring me already.
All right, let's.
Oh, somebody hold me.
We're gonna see.
More in science, I guess you could say that the 2 different species, I suppose, or I'm going to say 2 different kinds. All right, because okay, for the theory of evolution to work, let's just use the word species. All right.
Oh yeah, let's use the actual fucking terms in the field. Why not? Right?
Yeah, all those technical terms, you know.
Like,
Yeah, species is technical.
Like this is a problem. Like if he has to find so many filler words that aren't even related to the field, like why? Like why are you working harder than you need to when the information's already there? Like it's only a problem when you try to straw man what's there and create your own narrative. That's why he's having a hard time finding words, because you can't say that's a kind. Like what fucking kind is that? What kind is this one? What kind is any of them? Like, come on, dude, just stop. Like, it's so much easier just to be honest and read the shit as it is. Like, I don't understand why he makes life hard on himself. And then he wonders why people go, "You're not going to win this debate." I don't agree people should do that. That's their choice, but still, it's like he's making shit harder for himself. Anyway.
Those poop at people and those poop at other monkeys. They don't peel their animals. They don't peel their bananas before they eat it. Eat the banana straight up.
Actually, all of them do. All of them peel it.
They peel the banana from the bottom.
They don't do a bunch of different things. So there are clear differences between a monkey and a human.
So how? Wait. Pause.
So
the difference between how we eat a banana is some kind of unique trait that should be like put on the cladogram here.
I guess
that because I can eat a banana from the bottom as well, and I often do because it's actually kind of cool. You just pinch the bottom and you peel it open, and
that's how you're supposed to actually eat it. And
but we all know most people just do it from the top. But
Steve eats them right off the vine.
I like them right off the vine. Actually, it's an herb.
I know
it's an herb.
I know it's an herb. It's a herb. There's an H in there, Steve.
It says H is silent.
It's an herb. You don't pronounce the H in herb. I don't like saying herb. It doesn't make sense. Herb.
So for those of you who don't know what a cladogram is, everything along this... It's a what?
It's fruit. Bananas are fruits.
Okay, yes, bananas fruit. This cladogram, folks, like if you ever see one of these, everything along this line is related. Like everything here, the things here are more closely related to the ancestral line, which is somewhere back here. But as you move along, you have a divergence all of these have similar traits. You have a derived trait here for this, derived trait for that, derived, and so as you move along, you have specific sets of derived traits that allow them to branch off, and that's where you see these branching off things kind of here, these lines. But they're all related because they all have this will be, I guess you could say, the ancestral trait. Okay, that's why they're all related to one another. So
and Tyler points out that bananas are berries too, by the way. Yeah, that's true too. But they're also herbs and they're also fruits.
So you can have one more classification for a thing. That's possible. So there you go.
Monkey go from being this.
Yeah, only one classification. Yeah.
How does a monkey go from being this?
See this is what I'm talking about. See, this is exactly what I'm talking about. He doesn't know how to read this. He thinks that this turns into this, and this turns into that. That turns into that. That turns into that. That turns into that. That's not how that works.
He's thinking some kind of linear, like, change. No, these are branches for.
I knew he was going to do that.
G-man.
Folks, this is what I'm talking about. When he's upset that people point out he doesn't have an education, this is exactly what we're talking about. That is not how you read a cladogram. That's not how you're supposed to read this in any way, shape, or form.
Yeah, was he thinking that monkeys turn into humans and chimps turn into humans? Well, actually, are from monkeys, but he thinks chimps just and bonobos, they just turn into humans. Is that how he's reading this?
Yes, it looks like. So let me rewind it just a little bit. That's what I heard, that he's reading the monkey turn to the gibbon. So let me replay it just in case.
And how they sleep, they don't wear clothes, they don't do a bunch of different things. So there are clear differences between a monkey and a human.
Okay.
How does a monkey go from being this?
Well, what type of monkey? We are monkeys, but the monkey is a group, G-man.
Right?
Monkey is not a species, it's a group.
There's a difference between monkeys and technically, folks, we're great apes. We're classified into great apes.
I just want to point something out here. If G-Man's going to make his banana eating comment as a difference between one thing and another, I would just like to point out that everything in this diagram throws shit.
Yeah, everything here has the ability to throw shit.
We're all feces here. Yeah, we're all feces.
So we're all related.
Like this is painful to listen to. Like this is the shit that I'm taught. This is an exact reason why we say this. That's not how you fucking read this at all. If you would have actually looked up what a cladogram is, like looked up what this is called first, like actually gone through the Smithsonian website, something, and find, oh, we have this cladogram here showing, okay, well, what is a cladogram? Oh, how am I supposed to, oh, these are ancestral, this is the ancestral trait, these are derived traits. Right. Oh, okay.
Exactly. Let me try to explain this so G-Man will understand. The long line that ends with the person at the end is a street. The green dots are bus stops. Everybody's riding the bus, and when the gibbon got to its bus stop, it got off and went down the other street.
That's not actually a bad analogy.
But then you just keep riding the bus and you get to where you are, the human at the end.
Keep riding the bus.
Right. Everybody got off at their stop.
So is this an analogy that G-Man has to ride the short bus?
No, he's riding in the little trailer pulled by the golf cart behind the short bus.
Oh my goodness.
That's actually not a bad way to think about it. Yeah, but it's actually, like you said, derived traits, right? I mean, if one way to falsify evolution would be find traits that existed prior to what we would normally find, right? For example, like amniotic eggs. If you found that in somewhere in like 500, 600 million years ago, well, that would be a problem because they didn't have that characteristic yet. That trait wasn't around.
That's a derived trait.
It's a derived trait. So that would be a huge problem trying to put that into the evolutionary narrative, right?
Yeah.
Like how did hair come around before they had like chitin, or how did it come around before you had the erector muscle, or all these things are traits that build into what we are now, but you can't have certain things before other things because they're built upon existing homologies. And I think G-Man he's never understood that concept. Just never has.
Oh, there you go. Yes, thank you, Tyler Durden. Yes. Find a squid with an amniotic egg, that would be a problem. Thank you, Christopher. You're right. That would be a
problem.
That's definitely a derived trait that doesn't exist down that.
Find a trilobite that laid eggs.
Oh God.
Untitled by the late dinosaurs. Go ahead.
Let's see. CRISPR case. All right.
To being a gorilla or being related to a gorilla. Small changes over time. Okay. I don't know if this is the best chart in the world to use.
It's a pretty good cladogram. It's good enough for what you need it for. Yeah, it's not good because you can't read it.
It's like I said, this is kind of goofy, the one here, but I want people to get my point. In order to get here,
this is fine. This is perfect for what you need it for.
So in other words, a dog will have to become a non-dog. Okay?
No, you wouldn't be a fucking dog. You would see wolf here. Actually, no. There's before wolf. Shit. What are they called?
Dire wolf.
Carnivores.
You would put carnivore back here, but there's something that came before wolf that you would put down here, and then you could show how branching off. Well, you'd only have like a few branches, but you could show wolf, and then actually no. Now I think about it, because all dogs are derived from wolf. They have the same ancestral trait, but actually no. Yeah, you'd have to redraw
Carnivore morphians.
Whatever the fuck they're called.
The carnivore morphians are the ancestor to cat and dogs. That funny little creature, the myocid, that they discovered. It was like 50. I think it's 55 million years ago. But I mean, that's like probably no. But I think it's 55 million years ago. They found that myocid that was actually in the original clade of Carnorvia morphin. Wait, why do I have a hard time with this word? I'm trying to think it. Maybe let's look it up. Hang on. God damn it.
Instead of guessing, I'm just gonna look it up.
Carnorvia morphian. Yeah. Carnorvia morphian. And that was the ancestor to cats and dogs and bears. It says here. I just looked it up. 55 million year old fossil.
Myocids.
Yeah, myocids.
Mm-hmm. Myocids. Yeah.
Yeah, they were boreal. Most of them are boreal.
I'm trying to find a good example. I'm trying to find an example of a good cladogram for dogs. Dog bear. Man
bear pig.
Man bear pig. It could have been a thing. Man bear pig could have been a thing. You never know. Huh? Yeah. That's the tree one. I didn't want the tree. I wanted one that branched off like the one he has there. Anyway, that's okay. Anyway, moving on. Let's moving on.
Yeah. What's more dumb fuckery we have here?
Oh, the fox wolf would have been the first one, according to CRISPR. Fox wolf. All right.
This is a monkey. As eventually, after all these changes, according to evolutionists, over millions and 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 millions of years, going through a whole bunch of mutations, environment changes, eventually you're going to get a human being, right?
That's not how that works.
All these changes. How is the comment I'm making about a monkey becoming a non-monkey going against evolution?
Because that's not how you read a cladogram.
Because all monkeys, anytime you're a monkey, anything descended from a monkey, monophyletically, big word that he doesn't understand, is a monkey. That's why all mammals will actually have mammals. A mammal can never produce a non-mammal. A eukaryote can never produce a non-eukaryote. A tetrapod can never produce a non-tetrapod, which is why snakes, even though they don't have 4 limbs any longer, are still tetrapods.
I wondered, like, did he not read the stuff down? Because I'm looking at the Anthropodia here from where the monkey is, because it actually tells you, well, this is where monkeys and apes came from. And then you have here the hominidae. I can never pronounce that. Hominidae.
Some people say hominidae, some people say hominidae. But I think it's actually supposed to be hominidae.
Actually, there's a typo here too.
Because the other one's Homoidea, right? The bigger one, the big classification for all apes is Homoidea, and then the subgroup for the greater apes is Hominidae, and then the lesser apes would be Hylobotati.
Yeah, I'm not sure if they have. Okay, yeah, they go right into the great apes here. Yeah, they don't show any of the other one because it goes to Hominidae and then Africa. Is there, I guess, New World? They don't have New Worlds on here, do they? No, they don't. It's just old world monkeys. Okay, so yeah, for the great apes, you just have all the old world monkeys, and then it shows actually human lineage and the chimp lineage split, and shows a big ass red dot there.
Yeah, Limit says it's not Hominidae. I've heard it pronounced either way, Hominidae or Hominidae. I've actually, depending on what video you're listening to and what school, what pronunciation they're using, I've heard them pronounced both ways. You know.
Oh, that's so. Regardless of pronunciation, folks, this actually shows where it actually says here it's split. So I'm not sure if why. Like, yeah, it actually says split here. It says a linear split. So why would he assume that this becomes that? And it explicitly says so here.
I think it's day 2.
Big ass red dot.
CRISPR says it's day, and I usually pronounce it as Hominidae, but I've actually heard people in videos pronounce it Hominidae. But I the way I'm reading it pronunciation wise, I think it's Hominidae. So I'm just gonna go with that. Fuck it, whatever. That's how I say it, Hominidae.
Oh, check out this cladogram that
no idea.
That Lamed student produced. Check out this shit. Oh, that's nice. Fur, carnivorous teeth, protectable claws. Nice. Yeah, look at that shit. So somewhere in here,
characteristics.
Somewhere in here, a what are those things called? Not emus, the vicious ones.
Oh,
you know what I'm talking about?
Yeah, yeah.
The ones that'll fucking rip you open.
Oh man,
the terror bird.
No,
it is a terror bird, but it looks like an ostrich, but it's like a modern day velociraptor.
Yeah, no, I just saw a picture of one in a car the other day.
You know what I'm talking about too. Fuck, what are they called?
How old is this thing?
They've been around as long as ostriches.
Are they still living or dead?
Yeah, no, they're extant. Yeah, no, they're extant.
Yeah, I know it. It starts with an A, and I can't think of like
people keep them on ranches, but they can't. You can't go into the pen with
little fuckers
because they will literally gut you because of the claws on their feet.
Yeah, they have these big old fucking talon type things.
Very aggressive. Very aggressive.
I know. It starts with an A. I know exactly what you're talking about.
What are they called?
I hate that when I do that.
Shit.
Come on, guys.
You know what I'm talking about. Somebody, is 30 of y'all watching. What is that thing called? It's a fucking giant ass bird. It's as big as an ostrich, and it will kill. They have been known to kill people.
Swans. No.
No. No.
They've been known to kill people.
Oh, there's I looked up the kiwi.
Oh,
no, the kiwi's not that.
No, I know they start with an A. It's AGG something or other, and I can't remember what the hell they're called.
God damn it. Oh, come on. Someone knows what this thing is called. No, it's not. It's not an emu. They're like emus. A moa? No. They're not emus.
Not emu. No, it starts with an A. It's AGG something or
other. They're
a
lot like emus. I think they're related.
They're kind of like an emu or kind of like an ostrich, but they're alpaca.
No, it's not alpaca.
That's a llama.
Yes, a llama.
That's not alpaca.
The moa kind of looks like it, but with less fur.
God damn! I know exactly what I pictured it in my head. I thought it was starting with an A. I guess
it's about the same size as a moa.
I remember their claws too.
All right.
Shit, this is going to bother the oh god.
Yeah, they don't live next to me, so I don't have any of them to even remember what the hell they're called.
Stupid browser.
They won't even have them in zoos because they're so vicious. No, it's not a moa.
Stupid browser.
Damn it.
Yeah, we're gonna have to.
Cassowary.
Oh.
Yeah, I was gonna say cassowary, but someone said they started with an A.
Yeah, I was thinking I was thinking of alpaca, but it wasn't alpaca. But yeah, okay, cassowary.
No, they're cassowaries.
Yeah.
Yeah. World's deadliest bird.
They look like a flat turkey kind of fucking thing.
Yeah, they're not as tall as ostriches, but they're vicious, incredibly vicious. Yeah. Okay, so all right.
Yeah, no, cassowary is like an ugly fucking turkey.
Yes. Order the food. Order the food, dear. Oh my goodness.
It's a crocoduck, yeah. Cassowary is a crocodile.
Yes, it is crocodile.
Yeah.
Non-dog over a long period of time against evolution. You're telling me after 1 billion years, a monkey is always going to be a monkey? Well, if that's true, why is a tetrapod not always being a tetrapod?
Oh, God. All of these things here are tetrapods. Every last one of them. Every last thing on this fucking list is a tetrapod.
Yeah, he doesn't get that.
Oh, God.
You understand what I'm saying? The dishonesty that we see with people in the scientific community is really giving science a bad name.
Or is people who are scientifically illiterate, who don't know about the field, who purposely misrepresent it, and really don't. Like, this is funny. This is why I was highlighting the shit the other day when I was in a hangout with him and fucking Fiona and Zulu, that he's upset when people talk about the Bible and haven't read it, but yet he's saying all this shit and he doesn't even know what he's looking at, but he's making all these proclamations. So it's like that hypocrisy to me is just hilarious. Like, no pun intended. Like, I don't understand how you can, on the one hand, hold people to a standard that you yourself won't uphold. So there's just a little bit of education that needs to happen before he should have made this video. Like, I'm sorry. If you don't know what a cladogram is, look it up. If you don't know how to read it, look it up. These aren't terribly hard things to figure out on your own.
You know what, Drag? On South Park, they did an episode with evolution, and Mr. Garrison, or rather, I believe it was Mrs. Garrison at the time, explains evolution to the kids of South Park and uses such terms as retarded frog and like retarded frog, fish, squirrel thing. And, you know, that was a far better example of how evolution works, what G-Man comes up with.
Yeah, it's close enough. Shit. But
this literally was this thing fucks this thing and has a retarded baby, and then that one has like another retarded baby with another one, and that's how, like, it's a lot better than what G-Man comes up with.
I'm willing to give South Park the benefit of the doubt. At least they're somewhat educational.
Yeah.
All right.
If the first animal that scientists want to talk about, like where all life came from, and you want to say it came from a tetrapod and whatnot. Not all life, where everything things that we evolved from came from a tetrapod. Then a tetrapod has to produce another tetrapod.
And we're still tetrapods.
Yes.
This has to happen forever. Because
yes.
if it doesn't, then it's not evolution.
Yes.
Becomes a non-dog over billions of years.
It's still a dog.
The example that I gave in this video, how does that make any sense? That is a form of intellectual dishonesty that turns a lot of people off.
What is he talking about now?
This topic.
Hang on. He just literally said that tetrapods have tetrapods because they have to always be tetrapods, and now he's back on, well, dogs can have non-dogs. Oh, God, it's fucking stupid. Just fucking stupid.
Oh, God. Okay.
You're going to be debating this guy. I've just, for fuck's sake, dude.
Yeah. Yep.
You have my sympathy.
Yep.
That's what they believe. See what you guys are saying in the live chat.
You're saying you're an idiot, G-Man. Well, the 3 people you don't blocked.
Chat is disabled for this live stream, so I'm not sure. Oh, actually, no. This.
Oh, because it's after the video. That's
yes,
why.
after the fact. Yeah. So. Yeah.
I want to remind people regarding this channel, religion. We're not talking about that here. We're not talking about.
I don't know what we're talking about.
We're just talking about this topic of evolution.
I'm gonna fucking tell you.
Right? And I'm trying to do it in a way where everybody can understand. I'm not looking to bore everyone to tears. How you doing, Renee? Okay, she wants to see the chat in a minute. Shanny's watching.
Oh, Renee is in our chat too.
Yeah, I thought
Renee is pretty smart for a girl. I mean,
she can boil water.
Yeah.
She knows how to boil water. I have given it, and you know what? I have found out she's a very good sandwich maker.
What?
Yeah, pretty impressive. And she learned what? She learned that skill on her own. Pretty impressive.
No, she did. No, she didn't. She found a wiki how.
Yeah, she might have got help.
Hi, Renee. You know, I share a common ancestor with you according to evolution. Can't wait for that debate. So anyway, see that right there.
Oh. I should have just kept doing my chemistry homework.
I know. before. So, a lot of people will just turn people off from the topic because we all know that's what evolutionists believe. And at some point, over billions of years, these monkeys are not obviously going to be monkeys no more. And it's the claim of evolution. Okay, and in order for them to get out of that burden to try to prove that that's true, what many people will tend to do is that they'll just, oh, this guy, he's not good enough to argue DNA. He's not good enough to argue RNA. He's not good enough to argue taxonomy. He's not good enough to argue. So I'm just going to throw this out there. If a dog becomes a non-dog, then that's not evolution. So I just felt like making this video here a little bit to talk about this.
Nobody felt like watching it.
You gotta listen to people when they talk.
No, you don't.
Irony or projection here.
No, you don't.
Say short term. What does he say long term?
I think Tyler has the right idea. I think during the debate with G-Man, you should just do the whole thing. You should just be tuning your guitar the entire time.
Yeah,
I'm okay with that.
That's enough of this shit. That's enough. Okay, that's enough of this shit. That is so. Oh man, jeez. He likes to. Okay, he likes
it. Really hurts.
Just that is such bullshit. Oh, so
It
when
hurts.
he doesn't get your fucking point straight, and you correct them, it's semantics. But he's listening to you. Okay. All right. So on that note, folks, thanks for watching. My head hurts. I want to finish up a little bit more of this chemistry homework, and I'll probably just listen to some YouTube videos, some music videos, see what Mrs. Dragnut's doing, and
I got nothing planned tonight, so I don't even know what the hell's going to go on tonight. I'm just going to be working on this math presentation.
Yeah, if there's something going on in GDC, something interesting, just tag me so I can watch. Not necessarily, Yes, Chem 2. I am doing Chem 2. I had my homework up earlier in the Hangout. It's like we're going over like Henry's Law. Like it's nothing complicated. It's just reformatting the formula with I think they have a table up there, and I'm on my Pearson lab doing the homework. So yeah, I mean it's not anything incredibly difficult. Trust me, physics put me through the ringer and prepared me for these, so I'm not concerned. It's just I just want to take enough time to dedicate because there's a certain score I want to get on the homework. So that's really what it is. So Orgo, yeah, I look forward to Orgo. I think don't raise cassowaries. Okay, I'd stand corrected on the cassowary. People don't farm them because they are dangerous.
And their eggs are green.
Are they?
Yeah, their eggshells are like it's not like the eggs blend in with a nest or on ground cover. They look like giant green fruit.
Green eggs and ham.
Yeah, they're green.
Yeah, my chem instructor is Dr. Larag. He's pretty interesting, pretty young. I think he's maybe 5 or 6 years older than me, if that. Like he seems pretty young.
Can you imagine if the Living Dinosaur did a presentation or just a hangout on functional groups or something? How cool would that be?
You should get him to do it. Like we need to, guys. We should have had him here read this fucking paper because isn't he?
Yeah, this is much more along his lines. I'm sure. I mean, yeah, he's no joke. He's definitely a legit biochem dude. But I mean, I doubt he knows as much as Ronnie. But
nobody knows as much as Ronnie.
Nobody. That's impossible. No. How dare you even suggest?
Greg, what's Ronnie's full title? Is it Mr. Dr. Professor Ronnie?
Yes, I think so. I think that captures it pretty well. Okay. All right. I'm going to end this. Y'all have fun.
Thank you, Greg. Sorry, I got here late. I was busy.
It's fine. It's fine. I just kind of felt like doing it. Like it was one of those spur of the moment things where Sam contacted me. Hey, I got this paper. Want to talk about it? Yeah, sure. Why not? So, yep.
Yeah, I missed most of that. I saw it for a few seconds, but I was like, yeah, I have no idea what the fuck this is going on. I was honestly, no offense. I had no interest in the paper, so I didn't even know what the hell it was about or why you were reading it. Was it something? Was Nephew? Is that why? Did Nephew bring this paper up?
Oh, remember how Nephew always claims there's no evidence for abiogenesis? Well, guess what this paper goes over?
Oh, okay. I still had.
It doesn't mention abiogenesis specifically. It just talks about the peptide chains and how they can self-replicate.
I know what peptide is, but that's the only thing in that thing that I Yeah.
It goes.
Peptide is a small chain, smaller than a protein. It's only like 100 amino acids or less or something.
I don't know the number. I just know it's small.
It's a small
chain of amino acids. So the issue is, how do they get over the issue of not having any enzymes to help any catalysis happen? And so that's what it goes over basically. So the paper, if you guys want to use it, if you guys want to try and read it, be my guest. Paper is in the description, and you guys have a good night.